Besides, what have I got to offer? One thing you can't cook. I'm serious, Johnny. She's a highborn lady. So what?
Yes. Thank you all for coming in. People that are presenters have to sit on this side. There's a yellow thing that shows you where you need to be. For newcomers, people that are presenters have to sit on this side towards the front. There's a yellow sticker indicating where your seats are. So, good morning, everyone. I'm Sophie Frango, and it's my pleasure to welcome you to the 2022 Research Day that celebrates and showcases the preclinical, translational, and clinical research done in our department. The UBC and its surrounding areas operates on the traditional, ancestral, and unceded territory of the Costa Salish peoples, the Musqueam, Squamish, and Salish Waytooth nations, and we further acknowledge our gratitude and respect to all First Nation communities on whose traditional territories the university builds relationships and operates within the BC. I would also like to extend my thanks and everybody's thanks to the administrative staff of our department, led by Margaret Koshi and Vicky Yao, for their tireless efforts in putting together the many different logistic pieces that make this event possible. I would like to go briefly over the the schedule that we have today. And of course, I will ask all the virtual attendees to mute themselves so that so that we don't have a problem with unwanted sounds. So immediately after the introductory slides, Dr. Yatham will give the State of the Department address, and this will be followed by the first set of lightning talks. Those are very brief talks that showcase research done by our junior colleagues. Then we will break for coffee, which will be served outside, and during that time, you will also have the opportunity to view the posters. When we come back in, we have our main lecture by Dr. Christian Schulz focusing on addiction, and then the second set of lightning talks before we break for lunch. Lunch will be in a little box that will allow you to decide where exactly you want to have to sit and eat if the weather improves, perhaps even outside, but also the lunch break is a great opportunity to view the posters and to vote on the posters that you like the most. Then we come back for the afternoon sessions where we have a preclinical and a clinical talk, respectively presented by Drs. Rosing and Villa Rodriguez. And then we round up with the announcement of the awards and closing remarks. This session, of course, is chaired not just by myself, but also by Trisha Chakrabarti and Tim Murphy, who will be moderating together with me different parts of this event. So a few logistical things about how to vote. So when voting for the lightning talks or the best in-person posters, you have to go to Slido. You can see the link here and also the event code, and click on live polls, and then you can enter your preference. We will ask you to include your name, which will only be seen by the administrative staff, and this is simply to avoid duplicate voting. For Q&As, those who are attending in person, you just raise your hand and we'll come to you, the moderator will come to you. And for those that are attending virtually, you have to use Slido again and go the same event code, and you have to go to the audience Q&A, and the moderator will see the questions and relate them to the presenters. And with this, I would like to invite Dr. Yatham to give us the State of the Department address.
Thank you, Sophia. Good morning. Can you guys hear me at the back? Perfect. Thank you. Well, it's great to see so many of you in person. I think it's been nearly 3 years since we held an in-person research day. And of course, for this year, we debated a little bit to try and figure out whether we should have another virtual research day or an in-person. And I think at the end, we sort of decided that we can't live in fear forever. And I guess with the COVID numbers declining, although some might argue with that, we thought that the benefits potentially might outweigh in-person research day. And of course, we've asked people, only those that have been vaccinated, to attend in person, and also if anyone has not been feeling well, we've asked them not to show up. And of course, we've had a few people with some symptoms decided that they would not show up. So we've taken all the precautions to make sure that this is as safe as possible. Now, before I tell you a little bit about where things are with the department, I do want to take a minute to acknowledge that our department and number of our distributed sites across the province are located on traditional ancestral lands of Squamish, Musqueam, Tsleil-Waututh, Sykes, Okanagan, Lekwungen, and Laytonay people. And we're of course grateful for the opportunity to work and play on these lands. If you recall, we released our strategic plan. Hi, Ellen. Okay. We released our strategic plan around the research day last year. And as you will see in a minute, despite the pandemic, we've actually made significant progress in a number of areas. As you all know, we have 4 strategic pillars as a part of our strategic plan, and those include research, education, people, and partnerships. And because this is a research day, I'm going to tell you mostly about the progress that we have made in the area of a research pillar, but I might touch a little bit about some of the other areas. Just some facts and figures. Despite the pandemic, we've actually done exceptionally well in terms of funding. And of course, as we all know, without funding, we can't carry research. So close to $43 million of research funding since 2019-2020 fiscal year. And about 11 million of that came from CIHR, and close to 5 million came from Health Canada. So we're doing quite well. We continue to have 2 tier 1 research shares in the department, and we are finalizing a recruitment for a tier 2 CIHR tier 2 position in the department. We have approximately $10 million in terms of our operating budget, which we use to support various activities of the department. So here are some of the highlights from the research perspective. Now, as we went around and consulted, are you hearing the echo? Alan? What happened to our tech people? Let's see if I move this away. What about now? Okay, good, excellent. So, as I was saying, when we went around various sites across the province for consultation for our strategic plan, a couple of things that we heard from our members are, one was many clinical faculty expressed an interest in conducting research, but had no access to research funds. And secondly, a number of younger faculty and some of our fellows also expressed that they had challenges in accessing funding. So to address those issues, we're actually going to be implementing stimulus research grants from the department. So these are funded by the funds from the department, and we're going to have 3 different streams. One is the clinician stream. So these would be aimed particularly or primarily at clinical faculty. And then the second stream would be for junior faculty and also fellows and postdocs, those kinds of category of people that belong to those categories. And then the third one is aimed specifically at research in the child and youth mental health. And the funding for this comes from the BC Children's Hospital Chair in Pediatric Mental Health, supported by Roberta Sosi, who is the head of Child Psychiatry Children's Hospital. So we've been able to find funding to support this initiative, and the announcement about and the more details will be coming out fairly soon. We of course want to enhance research capacity within the department, and we obviously are trying to pick some areas where we need to put in more resources. And as you all know, within psychiatry, there's been a lot of interest lately in terms of novel compounds such as CBD and psychedelics. So we are in the process of putting up some infrastructure to facilitate that research. We're also going to be collaborating with the Academy of Translational Medicine to develop some infrastructure, or further infrastructure, I should say, for translational research to aid discovery of biomarkers, conducting clinical trials, etc. Of course, we want to make sure that we train younger people so that we have them take on the faculty positions that become available in the department, because within the psychiatry at the moment, majority of the members are at a full professor level. So as some of us retire, we obviously need to have younger faculty that can continue to conduct innovative research and take on these positions. So we've been lucky to have support from Marshall's family, and they're here, Stuart and Sunny. Thank you for your generous support. We've been able to implement Marshall's fellowship and scholars program, and over the past 3 years, this program has funded 14 fellows and 30 scholars, and this year we expect to fund up to 6 more fellows. We'll also continue to recruit. As I mentioned, we are recruiting for a CRC Tier 2 chair in the early intervention area in psychosis bipolar. And we also were successful in the faculty renewal program, so we're recruiting for a translational neuropsychiatry position. So that search has been completed, and we're also recruiting for a position to be based. This is a tenure track position based at the Northern Medical Program as well. And of course, we recruited 3 partner track faculty members, including Roberta Sasi and 2 colleagues from Korea. Both are here, Dr. Kyosub Ha and Kim Soo Hong. We are implementing some changes to departmental structure, both in terms of divisions and programs, and you all have received communication about that in terms of the changes to the divisions and programs, and these will be implemented in the next few months. And those will include specific reporting structures. And the idea here is to really pull people with similar interests together to facilitate further research. The other thing that we wanted to ensure is communication across the department. Of course, with the pandemic, that's been a significant challenge. We haven't had too many meetings, but going forward, based on the advice from our advisory committee and also a number of faculty members, we're going to have an academic faculty meeting twice a year, and we're also going to have an advisory committee meeting twice a year so that the issues that come up can be communicated and also timely consultations can be achieved to implement any changes within the department. The other thing that's come up quite frequently is, again, this is from not only people in the community, but also from our own faculty members, is that if they want to find out what's a particular faculty member is doing in terms of the research, people have had significant difficulty with that. So to address that, what we've been doing is refreshing the departmental website, and the new website will be hopefully launched within the next 2 to 3 months, and this will have a much more modern feel and look, and we'll also have lots of information about our faculty, the research that's going on, and also information to general public, so that if anyone wants to participate in any of the research studies, they will know what the studies are about, and they can access information and maybe even contribute to research funding for any of the PI's labs as well. The department has also implemented an active social media platform managed by Margaret Koshi, and we're very active on Twitter. We now have up to 900 followers. Lots of research is going on in the department, and it'll take several hours for me to talk about everything that's going on, but I thought I'll just take a minute to highlight research from a couple of junior faculty members and a couple of senior faculty members. So you can see Daniel Vigo's lab has attracted a huge amount of funding to look at student mental health. And of course, Fidel Villa Rodriguez has been doing some innovative work with fMRI and transcranial magnetic stimulation. You'll hear a bit more about it later this afternoon. Some work from Todd Woodward's lab looking at how using the task-based fMRI to look at the neurobiology of delusions. And Erin Mihalik's work with CREST-BD has attracted attention from around the world in terms of some of the work that she's been doing to help improve quality of life for people with a bipolar disorder, for example. This is a long list of grants and awards various people have received within the department, and I won't go through the names, but as I mentioned, we've been incredibly successful in terms of grant funding. And lastly, as I mentioned, I'll touch briefly on the progress in other pillars. We've done lots of work, but just to give you a couple of highlights with the education, psychiatry has training programs at Fraser, Island Medical Program, and Northern Medical Program. So the only medical program where we don't have psychiatry residency is in the interior. So we've been working with the Ministry of Health closely over the past 2 to 3 years to make that happen. We don't have an official confirmation as yet, but we have information that suggests that we will get funding to expand residency training program to interior health as well, which is fantastic news. We're also working on implementing a master's program in translational psychiatry led by Ray Lamb. And again, we're hoping that this will be completed by the end of this year. A couple of things about addressing the concerns expressed by our faculty members. The clinical faculty, of course, are the backbone of the department. Without clinical faculty, we can't do most of the things that department does. And one of the things that we heard loud and clear from our clinical faculty is how cumbersome and tedious the application processes for appointments and promotions. So we're now changing that. We're streamlining that to make it much more simple, and that's going to be ready for implementation within the next 2 to 3 months. We've also been looking at the equity, diversity, and inclusion aspect, and of course, all of you will have received that survey. We got the feedback. We're now collating that information, and based on that, we'll implement plans to address any inequities in those areas. And lastly, in terms of partnerships, we've been building active collaborations, not only locally, but also regionally, nationally, and internationally. A couple of things to highlight are one is our partnership with the BC College, because as many of you know, there's a huge shortage of psychiatrists within BC. And so we've been working with the college to come up with the novel ways to address some of those shortages, including changing some of the licensing requirements, implementing fellowship programs and things like that. And also working closely with the ministry to increase residency training positions as well. So those are some of the highlights. And of course, much more is happening within the department. And I think maybe perhaps at the next annual departmental meeting, I'll give you more updates. But for now, I think hopefully that has given you some idea with regard to what the department has been doing over the past year or so. Thanks very much.
Hi, good morning, everyone. I'm Trisha Chakrabarty. I'm an assistant professor in the Department of Psychiatry, and I'm, along with Sophia and Tim, I'm one of the organizers of today's events. So I'm very pleased to introduce the first set of lightning talks. So this will be the first of two lightning talks that we have. So this is a format wherein some of our junior colleagues present their research in a small bite-sized 5-minute format. And you'll see that these talks give us a chance to really showcase the breadth of research that's being done. In our first set alone, we range from looking at neuromodulation in depression to the molecular bases of autism spectrum disorders. So each presentation will be 5 minutes. I will, just for the presenters, I'll give you a wave if you're going over. We are going to save the questions till the end, so please make note of any questions you have, and then we'll be able to address them at the end. And a reminder for our virtual attendance, you can put in your questions on Slido, but please make sure to indicate to whom your question is addressed. And I just want to flag a small change in the program. King Zhang was not able to join us today, so she submitted a pre-recorded video, which we'll be presenting at the end. So Natasha Verzosa will be going after John Jose Nunez. And unfortunately, King won't be able to take any questions, but if you have any, we can certainly email her and she can get back to you in that way. So our first presenter is Elizabeth Gregory, who is an MD PhD student and a member of the 9net lab led by Dr. Fidel Villa Rodriguez.
Okay, how do I switch the slides on it? Is it just...
Oh, perfect. Thank you so much.
Oh, okay. Okay, good morning, everyone. Thank you, Dr. Chakrabarty, for the introduction. So let me see. Okay, so today my talk will discuss individualized functional targeting for repetitive transcranial magnetic stimulation and its utility in treatment-resistant depression. Okay, so RTMS is an effective treatment for treatment-resistant depression, and it's typically delivered over the left dorsolateral prefrontal cortex, or DLPFC. And this is thought to be a good target because of its connectivity with the subgenual anterior cingulate cortex, which I will be referring to as the subgenual. And so the subgenual is abnormal in depression, and importantly, modulating the connectivity of the subgenual seems to be relevant in treating depression. So the subgenual is not accessible by RTMS, which is limited to cortical surfaces of the brain. And so one way of accessing it is by stimulating the DLPFC. And so the region of the DLPFC that shows the greatest anti-correlation or out-of-phase activity with this subgenual is thought to be the optimal treatment target when it comes to RTMS. This location does vary quite a bit between individuals, whereas the treatment target when delivering RTMS is typically done using the same anatomical location across patients. And so it's thought that creating individualized targets based on a person's functional connectivity can improve their response to treatment. And this has been investigated retrospectively in small samples, but it has not yet been validated in a large sample. And so to do this, we performed a retrospective analysis of individualized functional targets for 340 subjects who had received high-frequency RTMS to the left DLPFC. And the treatment target that was actually used in the study was the same for everyone using anatomical coordinates. And so this is indicated by the neuronavigated target that you can see on the slide. So patients had received 10 minutes of resting state scans before starting the treatment. And so we used these baseline scans to derive an average time series for the subgenual region of interest, which is indicated in red there. And we also derived time series for each individual voxel within the DLPFC, which is kind of this colorful mass on the screen there. And so the subgenual and DLPFC time series were then correlated with one another. And to identify subjects' optimal individualized targets, we chose the DLPFC voxel that showed the greatest anti-correlation to the subgenual. And to find the anti-correlation at the site of treatment, so that neuronavigated target, we simply correlated its time series with the subgenual seed. And so you can see in this image, what's depicted is the anti-correlations within an individual subject. So their individualized functional target, which you can see has a much greater anti-correlation with the subgenual compared to their actual neuronavigated target, which shows actually no anti-correlation with the subgenual, a slight positive correlation. And so we hypothesized that patients whose optimal target was closer to their actual treatment target would have better response to treatment. And we also hypothesized that functional connectivity at the treatment target would be associated with treatment response. So in other words, if we were hitting a region with greater anti-correlation, we would be getting a better response. So this slide now is showing the location of the actual treatment target in blue and the average location of the individualized targets in responders and non-responders. And so response in this case is defined as 50% or more reduction in depression scores. And the MNI coordinates of these targets are indicated in the brackets below. So as you can see, the average optimal targets for responders and non-responders are in fairly similar locations. And actually, when you look at the distance from these targets to the actual treatment target, they're no different. So in other words, responders and non-responders don't seem to have targets that are different distances to the actual treatment site. And when you look at response as a continuous variable, we did not find that distance between the individualized and actual treatment target seemed to matter for treatment response. So percent change in depression scores was not correlated with how close we were to the treatment site. And this also held true when controlling for variables associated with treatment response to RTMS. So that's indicated by the R prime on that figure. Okay, and then finally, looking at the anti-correlation at the actual site of treatment, we also did not replicate the finding that greater anti-correlation at the site of stimulation was associated with treatment response. So in all, we did not find evidence that targeting the individualized DLPFC subgenual connectivity seemed to matter for treatment response to RTMS in this large sample. So it may be that we got a good enough target using the group level coordinates, or that differences in analysis methodology when it comes to processing the imaging files are affecting our results. And so as further steps to confirm that our results are indeed null, we plan to explore other methods that other researchers have used to confirm that our findings are not biased by our methodology choices. Furthermore, we plan to look beyond the simple functional connectivity as a method of treatment targeting and include other parameters that are known to affect treatment response, such as looking at electrical fields elicited within the brain to further improve treatment outcomes. Thank you.
Thank you very much, Elizabeth. So next we have Riang Ji, who is a research associate and a member of Dr. Sophia Franco's research team.
Somebody needs to mute their microphone.
Good morning, everyone.
Just a reminder to everyone on Zoom, please mute your microphone.
Good morning, everyone. It's my great pleasure to talk about our work on behalf of our co-authors. The title of this work is Neuroimaging-Based Profiling Identifies Distinct Transdiagnostic Subtypes of Youth with Mood and Anxiety Disorders as a Transition to Adolescence. Adolescence is a very important life period, and we're very interested in the mood and anxiety disorders in this particular age range because there's compelling evidence that a huge amount of mental illness, including mood and anxiety disorders, began by age of 14 years old. And as we all know, those clinical populations are full of heterogeneous, and they have shown brain anatomical changes. So the aim of this study is to identify the clusters of youth with mood and anxiety disorders based on the multimodal neuroimaging markers. To achieve this goal, we use the ABCD study, which is so far the largest US-based brain developmental and child health cohort. In this study, we use because ABCD study is an ongoing longitudinal study. So in this study, we use their baseline data, and we picked the ABCD participants who have never had life history of any mental illness, aka the typically developing participants, and also we picked the participants only with history of depressive disorders, only with history of bipolar disorders, only with history of anxiety disorders, as well as participants with comorbid mood and anxiety disorders. So for all of those participants, we first extracted the multimodal neuroimaging multimodal neuroimaging phenotypes, including brain morphometry, intracortical myelination, and neurodensity in cortical and subcortical regions. Then we fed this neuroimaging markers or features into a machine learning algorithm called Hedra, which uses the typically developing group as a reference to divide the participants with mood and anxiety disorders into different subtypes or clusters. And then we compare those identified clusters or subtypes and compare them to the typical developing group in terms of their obstetric and prenatal history, cognitive and parental characteristics, family, school, and neighborhood environment. Next one is the result. For the results of neuroimaging, we found that the three cluster or three subtype represented the data best. So we stick with the three cluster solution. And we found that these three subtypes, they did not differ in the diagnostic compositions, means that they are transdiagnostic. And for the comparison between each subtype and the typically developing group, we found that the first subtype is very likely to have the accelerated brain maturation with use this. Oh, I cannot use it. With the globally greater reduction in this surface expansion and greater reduction of the thickness in the prefrontal and some of the parietal cortex. The second subtype shows the delayed brain maturation with the greater, with the more cortical thickness, higher cortical thickness and lower surface expansion, as well as the lower myelination, cortical myelination. The third group, subtype 3, showed a very atypical brain maturation pattern with lower cortical thickness, lower surface area, subcortical volume, and higher cortical myelination, higher neural density, globally, usually. And for the non-imaging factors, the comparison between each subtype to the typically developing group found that generally the three subtypes have very similar characteristics and adverse exposure, including more maternal alcohol substance use, more maternal medication, more internalizing and externalizing problems, decreased positive view of school, more parental psychiatric problems, more family adversity and conflict, less parental engagement or parental supervision, more advanced event, and more bullying. In particular, the third group, which is the atypical development group, this group showed the worst academic and school performance, and they lived in neighborhoods with lower socioeconomic status. So, summary. First, the neuroimaging-based machine learning identifies 3 clusters of youth with mood and anxiety disorders. The cluster were transdiagnostic, which reinforced the formal findings that the alignment between the clinical diagnosis and the underlying brain mechanisms is very poor. And then these 3 clusters showed very minimal differences in terms of the adverse exposures, although the clusters with the most atypical maturation pattern experienced a greater level of adversity. And as for the future direction, we'll keep going to assess the longitudinal effect of the clusters, and hopefully it will inform us about their evolution during adolescence. So thank you.
Thank you very much, Ruyang. Our next presenter is Ahmad Samara, who is a Master's of Neuroscience student and a member of Dr. Tamara van der Waals' research team.
Hi, everyone. Can you hear me? Great. I'm very excited to be here today, and I will be presenting on the macroscale cortical organization during naturalistic viewing. Okay, this project combines 2 emerging fields in neuroimaging. The first is naturalistic imaging, which are studies that use complex multimodal real-world stimuli to study brain function in order to increase the ecological validity of these studies. And the second field is the use of gradients to study brain organization based on certain features such as functional connectivity by describing the gradual change of these features across the brain. And these gradients has only been described so far in what we call resting state imaging, which is we ask the patients to lie in the scanner and not engage in any task and nothing in anything in particular. And here we wanted to ask whether these functional connectivity gradients are better characterized or has evolved to support the kind of processing we see during this resting state or during the more complex cognitive processes that we see during naturalistic imaging. In order to answer this question, we looked at data from the Human Connectome project, where young adult subjects were scanned during rest and movie watching states on a 7 Tesla scanner. We started by computing a functional connectivity matrix for rest and movie fMRI. And functional connectivity is a measure of the similarity of the activation of a pair of brain regions over time. And when we compute a functional connectivity matrix, we're representing this feature across the whole brain. And based on this functional connectivity matrix in rest and movie, we computed a set of functional connectivity gradients by applying a dimensionality reduction technique called diffusion embedding using the brain space toolbox. And we obtained a separate set of gradients for rest and movie. Now, if we look at the rest functional connectivity gradients, we got, they nicely replicate previously described gradients in the literature, where only the first gradients follow a hierarchical organization, basically going from unimodal, somatomotor, visual, auditory regions, all the way to higher order regions in the brain, such as the default mode network. When we looked at movie gradients, however, we see that all of the top 3 gradients in movie watching fMRI data are hierarchically organized, but they each separate out a specific modality. Somatomotor in gradient 1, visual in gradient 2, and auditory in gradient 3. Now, this organization in movie watching makes sense as a brain organization, but we also wanted to see if this organization also relates to behavior in a meaningful way. So we compared the predictions of behavioral scores that we get based on features from resting state and movie watching. And we see that movie watching gradient features are better at predicting cognitive scores compared to rest. Overall, we're quite excited about these results. We think that these modality-specific hierarchical gradients might provide a unique tool for studying brain organization in psychiatry. And because in many psychiatric disorders, we see that this hierarchical organization is disrupted. This is still an ongoing work, and for the next steps, we want to look at the reliability of these FC gradients in movie watching data, and we also want to look at the correspondence between these gradients, which are functional, with other structural features in the brain, such as gene expression. And more long-term goals would be to look at the gradient organization in neuropsychiatry and to hopefully contribute to efforts to develop biomarkers based on functional organization in the brain. Thank you.
Thank you, Ahmad, and bonus points for fitting that into 4 minutes instead of 5 minutes. Our next presenter is John Jose Nunez, who he's no longer a psychiatry resident because he just completed and passed his Royal College exams. So he's a psychiatrist. But he was in the research track, and his project was supervised by Dr. Alan Bates in the Department of Psychiatry and Dr. Raymond Ng in UBC Computer Science.
Thank you so much. No, not sure I passed everything. I was very lucky, but okay. Well, thank you so much for having me here today. And I'll just make sure this works. Yeah, perfect. Okay. So as you could probably guess, cancer not only impacts our physical health and mortality, but also leads to difficulties with our psychosocial health. And because of this, cancer agencies like BC Cancer will employ psychiatrists, counselors, social workers to help address these needs. However, it's well documented in the literature that the treating oncologists, the med-oncs, the rad-oncs, the surgeons, often have a difficult time identifying what patients benefit from referral. And this leads to some patients falling through the cracks and not accessing these resources. So I thought this might be a cool application for artificial intelligence, for machine learning, to automatically detect what patients might benefit from the referral. And this could one day lead to a system that maybe automatically pings the oncologist, hey, you should ask this patient the next time you see them about whether they'd benefit from a psychiatry referral or not. But of course, whenever we want to do machine learning, the prediction is only going to be as good as the data. So we had to pick a data source. We thought that the initial oncologist consultation document might be a good source of data for this prediction. They tend to be somewhat big documents, not quite like psychiatry documents, but somewhat long. They'll have like social history, family history, symptoms, function, that sort of stuff. And importantly, you see any patient across the country at any cancer agency, they're all going to have this document. So you could pretty much use it for any patient. So we thought, let's try using AI to make this prediction. And just for terminology, whenever we apply AI to like a text document to natural language like English, we call this natural language processing, or NLP, acronym we'll use. So how does this actually work? I thought we could do a bit of crowd participation, but a human intelligence first, these two little examples at the top. So we have Mr. Oski and Mr. College. You could probably tell what I was thinking about when I made these slides. So show of hand, you can read those two. If you think Mr. Oski would be more likely to need to see a psychiatrist. Yeah. So everybody's like, yeah, of course. You read that, it's pretty obvious. And now our AI is, using a whole consultation document. It's not as obvious as this. But how does the AI actually come to this prediction? So the traditional method is something called bag of words. It's on the left of the screen there. Essentially what you do is you just look at what words are in a document and you count how many times the words occur. You don't care about order or whether there's a negation or anything like that. It's kind of simple. It tends to work somewhat well, but it can lead to problems. You can see both of those little examples I made have pretty similar words, like what leave is in both, but one of it is, the wife leaving the patient, one of them is leaving to Cancun. Those obviously have very different impacts. The neural NLP methods, the neural artificial intelligence models, and neural networks have an advantage because they can actually care about the order of the words, they can understand words together, and so might actually realize that if you have wife right before leave, it's going to be different than you leave to Cancun. So we had the opportunity to develop and evaluate these models on a 50,000 patient database at BC Cancer. The models did work, I think, reasonably well. The balanced accuracy and the AUC were both above 70%, which is pretty in line with a lot of applications of AI in psychiatry. And nobody's really done anything like this before, so I think this is a good kind of an initial result. Two of the three neural network methods that we tried were able to outperform the traditional method, the convolutional neural networks, long short-term memory models, and these are two common networks that we'll often use. So yeah, happy to talk more about the results. I thought they were kind of fun. With my last minute, I just thought I'd show actually how the model is kind of understanding, because I think it's pretty cool. So at the bottom here, we have an actual excerpt from, very de-anonymized, very anonymized, of course, from one of these oncologist consultation documents. And this is for a breast cancer patient who did see psychiatry, and the AI was able to predict this. And this shows how the AI understood it after training on about 40,000 other documents. So reading all these documents, teaching itself how to make this prediction. I didn't tell it how to do anything. It read 40,000 documents and then tried to interpret this. And you could probably just make out, the little green there. So the green words are words that I thought was predictive of seeing a psychiatrist. And you could tell that a maternal grandmother with breast cancer was predictive of seeing a psychiatrist. The AI thought this meant the patient would have a likelihood of seeing a psychiatrist, which is kind of interesting, right? This kind of this generational aspect, perhaps having when she was young, she saw her grandmother suffering through the difficulties of breast cancer. And so when she was diagnosed herself, she was quite anxious about it, or something like that. I'm not sure. It also, there's a bit of a red shady there for non-smoker. So it also started to understand the connection between smoking and seeing a psychiatrist. So it thought that because she was a non-smoker, that was a slight negative predictor of seeing a psychiatrist. So it was actually able to teach itself the connection between smoking and mental illness that we know about. So that's everything. I'm happy to answer questions in the question period, and thank you so much.
Thank you very much, John Jose. Our next presenter is Natasha Rososa, who is a clinical research assistant and a member of the Majors Project, led by Dr. Robert Stowe.
Hi everyone, so I'm very excited to be here today representing the majors group. So today's talk is going to be on assessing the utility of pharmacogenomic testing in a cohort with treatment-resistant schizophrenia or schizoaffect disorder. So like I said, this is out of the MAJORS project, and MAJORS is a pilot multimodal omics and psychiatric genetic counseling project conducted in participants with treatment-resistant schizophrenia or schizoaffective disorder. So we've completed whole genome sequencing on our first 25 participants of the cohort, and we were wondering and wanted to assess the relative frequency of poor, intermediate, rapid, and ultra-rapid metabolizers at the drug metabolizing cytochrome P40 CYP enzymes, most clinically relevant to commonly used psychiatric medications. So that would be CYP2D6, CYP2C19, and CYP2C9. And we hypothesized that pharmacogenomic differences might contribute significantly to our cohort's poor response or intolerance to these agents. So this little pie graph donut hole here, so in our cohort, participants are taking 136 medications, and 19, or 44%, had an associated recommendation. Of the 3 CYP enzymes, CYP2D6 accounted for the majority of PGX recommendations, and 13 pertained to CYP2D6, whereas 5 pertained to CYP2C19, and 1 to both CYP2D6 and CYP2C19. This bar graph depicts the results of the MAJORS cohort phenotype. Sorry. For some reason, I don't think the bar graph is showing up properly. That's a little bit unfortunate. But anyways, what the bar graph was supposed to show was the results of the MAJORS cohort phenotype frequencies for the 3 CYP enzymes when compared to the maximum estimated phenotype frequency across all ancestral populations. So the ancestral populations that were supposed to pop up were the African, African American, North American, European, East Asian, South Central Asian, Latinx, and Oceanic distributions. And also supposed to pop up on the bar graph was that at CYP2D6, 8 participants out of our 25 were poor metabolizers, 13 were intermediate metabolizers, and at CYP2C19, 1 participant was a poor metabolizer, 5 were intermediate metabolizers, 2 were rapid metabolizers, and 1 was an ultra-rapid metabolizer. And at CYP2C9, 1 participant was a poor metabolizer and 6 were intermediate metabolizers. And what was really interesting and notably was that our cohort had significantly greater CYP2D6 poor metabolizers at 32% versus 6.5% in the general population, with a Z-score of 6.3 and a p-value of 0.0001. So in support of our hypothesis, we found an enrichment of CYP2D6 poor metabolizers in our cohort, and that's a metabolizer phenotype linked to an increased risk of psychotropic drug intolerability. So our findings support the notion that PGX testing for CYP2C19 and CYP2D6 may be most beneficial when there is a history of adverse reactions to multiple psychotropic medications. And we thank our donors for their generous support and our participants for their generous participation as well. Thank you very much.
Thank you very much, Natasha. So next we have our pre-recorded presentation from King Zhang. Just a reminder before I start that for our virtual participants, you can start putting your questions into Slido, and then we'll be able to get to them during the question and answer period. So I'm hoping.
Good morning, everyone. I'm Qingdong from Dr. Wei Hong Song's lab. It's my great honor to present part of my thesis project here, titled Cleavage of CanSNAP2 by Furan, Atom 10, 17, and the gamma secretase, and its effect on autism spectrum disorders. First, some backgrounds. CanSNAP2 is one of the largest genes in the human genome. With its variants increasingly found in ASD patients, CanSNAP2 has been considered as a critical target in ASD. In animal studies, CanSNAP2 knockout mice and rats show ASD-associated phenotypes. However, the mechanisms underlying CAN-SNAP2 disruption in ASD remain unclear. Our research focuses on the proteolytic processing of CAN-SNAP2. Why is proteolysis or cleavage so important? Here are two examples. Notch receptors undergo sequential cleavages by furin, alpha, and gamma secretase to release an NICD. The cleavage is critical for development, and its disruption causes death in the embryonic stage. APP is cleaved by beta and gamma secretase to produce A beta, which forms the amyloid plaque in Alzheimer's disease. Therefore, proteolytic processing is critical for protein function. In a separate study, we discovered that CAN-SNAP2 is cleaved by gamma secretase to generate an intracellular domain CICD, which rescues ASD-associated phenotypes in CAN-SNAP2 knockout mice. Here, we hypothesized that CAN-SNAP2 can be cleaved by other proteases upstream of gamma secretase, which may be impaired in ASD. To review the pathway, we asked 3 questions. Who cleaves CAN-SNAP2, where are the cleavage sites, and what is the order? We used overexpression and knockdown experiments to reveal that ADAM10 and 17 dependent alpha secretase cleaves CAN-SNAP2 to generate 2 CTF-alpha. Furin also generates a CTF-F. CAN-SNAP2 on the cell membrane was cleaved. Next, we identified alpha cleavage sites using mass spectrometry, N-terminal sequencing, and CAN-SNAP2 protein ladder. Results from 3 methods all identified that ADAM10-17 primarily cleave at I79 to generate C79 and less at L96 to yield C96. We further asked about the order of cleavage. Transfected cells were treated with ADAM17 and furin inhibitors. The 2 inhibitors showed opposite effect on upstream CTF-F, while both reduced downstream CTF-alpha by different mechanisms. Through more experiments, we revealed that CTF-F generated by furin is further cleaved by alpha secretase. C96 is cleaved to yield C79, and C79 is further cleaved by gamma secretase to generate CICD. Since CICD can rescue ASD phenotype, we further asked whether CAN-SNAP2 cleavage is impaired in ASD. In a previous study, several point mutations in CAN-SNAP2 were identified in ASD patients. We discovered that I1253T at alpha cleavage sites corresponding to the C-terminal I79 abolished the alpha secretase cleavage to generate C79. Other mutations showed less maturation and reduced C17 levels. Furthermore, alpha secretase cleavage is impaired in overexpression experiment. This result demonstrated that ASD-associated mutations impaired CAN-SNAP2 alpha secretase cleavage. In summary, we discovered that ADAM17 and 10 cleaves CAN-SNAP2 at C-terminal L96 and I79 sites. CAN-SNAP2 undergoes sequential cleavages by furin, alpha, and gamma secretase. C79 is required for gamma secretase to generate CICD. In a separate study, we found that CICD can rescue ASD phenotype. Here we revealed that the upstream alpha secretase cleavage is impaired by ASD-associated pathogenic mutations, which may underlie ASD pathogenesis. Thanks to my supervisory committee and funding source for supporting the study. Thanks for your attention and welcome questions.
Okay, so thank you to all of our presenters. It takes a lot of skill and courage to try to communicate your research findings in 5 minutes, and I think they all did an excellent job. So at this point, we'd welcome any questions from our audience. And again, for our virtual participants, please put in your questions on Slido. And for our on-site participants, if you're asking a question or answering it, please just make sure to turn on your mic so we can pick it up. So were there any questions for any of our presenters?
Sorry, can you hear me? Can you hear me? Yeah, okay. So I have a question for Mr. Ahmad Samara. And I was wondering what tests he used to assess cognitive function in his research, where he associated the neuroimage with the cognitive function. So what tests you use it to assess these cognitive functions?
Oh, great. So that was a question for Ahmad Samara about the test that was used to assess cognitive functioning that he mentioned.
Yeah, can you hear me? Okay, so we didn't use any task here, and we're actually looking at these functional gradients during movie watching, which is a more naturalistic brain state. So instead of engaging in a very specific task, for example, language or math or seeing specific photos, we're just presenting the subjects with a full clip from a movie. And that's the state we're studying. And we're comparing this to resting state, which is any scanning session that doesn't involve any specific task or any specific thinking processes. Thank you.
I think I just had a follow-up question to that. I think you said that you found that the functional gradients were correlated with behavioral measures and cognitive measures. So I think the question was what exactly you used to assess those behavioral and cognitive measures.
Oh, so the same data set we used includes separate behavioral measures that were obtained using questionnaires from established tasks. And these tasks or these questionnaires fall under specific domains. For example, cognitive scores or emotion scores or motor scores. So we looked at these specific separate scores based on certain domains. And these are not obtained during the scanner. They're separate scores for the subjects that underwent the scanning sessions.
But that's where you saw your imaging behavioral correlations.
So the idea is that we use data from the scanning sessions in order to predict the subject scores that were obtained outside of the scanner based on their connectivity patterns.
Yeah, thank you.
Thank you.
Were there any other questions?
I have a question. Hi. So for the second to last talk, does the poor metabolizing phenotype at CYP G6, I think it was, does that predict lack of benefit of the medication or more intolerable side effects so the medication is stopped? Just wondering if there are, it's more one or the other. It's Dr. Verzosa.
There we go. Right, so that's a good question. So it was for CYP2D6, and what we're doing is we're looking at the pharmacogenomic reports, and then during the results disclosure, if there is an appropriate recommendation, we're letting the participant or the patient know whether or not a recommended change in the dosage would be a good idea. So
Natasha, I think we're having a hard time hearing you.
Oh, sorry.
If you
maybe
just
if I lean in a little bit
pull
more.
down your mask and speak into the mic.
Here, maybe I'll try removing my mask as well. Is that a little better? Okay, so for CYP2D6, it was the poor metabolizers. And what we're doing is looking at the recommendations from the pharmacogenomics reports. And during the results disclosure, and for the results disclosure appointments, we are talking to the participants and letting them know if there are any actionable recommendations, and they can take it to their psychiatrist or their pharmacist, and appropriate drug dosages can be made. Drug dosage changes can be made.
Thank you. Were there any other questions?
I have a question for Elizabeth Gregory. A great talk. I like the idea of the of the personalized treatment. I guess I was wondering about the resolution of TMS. I thought it was relatively coarse. Oh, and it seemed like the distances you were measuring were, I don't know, like 10 to 40 millimeters. So is it really possible over this small scale? Like if you could image the actual activation, would it be like the whole, I don't know, frontal cortex or something?
Yeah, that's a great point. So that's kind of why I want to bring in the electrical fields at some point to understand how much of the cortex is being stimulated and whether these differences of a couple of centimeters matter. So yeah, the distances varied from about basically right on target to 5 centimeters. And so an important piece too with my data is that our study used a cohort of subjects that all got the same MNI coordinate. So they were all targeted in a very similar part of the brain, whereas other studies that have found that this distance feature seems to matter used scalp-based measurements. So basically like 5 centimeters from the motor cortex, for example, which is going to look very different depending on your head shape and size. So I think, yeah, to your point, we're probably hitting close enough to most people's targets. And the TMS field is probably a couple centimeters or so wide. So yeah, I think that's an important consideration. I would like to look into it more to understand with the electrical field, but yeah, I think that's probably contributing.
Thank you. Any other questions for our presenters?
Hi, this question is for John Jose Nunez. I was curious for your machine learning model, how did you validate like the predictions were correct? Was it that like you used data where you knew that people ended up seeing psychiatrists, or I was just curious how you validated it.
Yeah, that's an excellent question, because of course machine learning, I mean, has such a dependency on how you actually evaluate your results, whether it's valid or not, right? Because you could do totally weird stuff, and oh yeah, look how awesome this is. So in this case, we just had a holdout set of patients, and for all 50,000, we knew whether or not they were to see a psychiatrist. So yeah, we just had a holdout set with the models hadn't seen before, and saw whether or not it could accurately predict on that set. Thanks for the question.
And I just had a follow-up question for John Jose. I was wondering, so you looked at seeing a psychiatrist as your outcome, but I was wondering if you had a chance, or if you're aware of other work that's looked at actual like treatment-related outcomes using this kind of NLP AI.
Yeah, not really. There's actually like very few papers, especially using like neural NLP in psychiatry on clinical documents. There's obviously, stuff using like structured data, like gender and age and that sort of stuff. But in terms of like using clinical documents, it's definitely kind of a pretty new field. A lot of it depends on like graphics cards, like the actual computer parts, because we need like really cool hardware to actually run this stuff. So it's all pretty new, but I think there could be some really cool applications of it for sure.
Thank you. Any other questions?
So a question for, let's see, it's Ruyang Ki. I think you did the talk about predicting mood disorders or mental health disorders in youth. So I just wondered if after training your AI algorithm on the data you had, did you try using an independent set to validate it? And also why choose age 9 to 10? Why that window of age group?
Oh, Dr. Raymond, if you could just turn off your mic.
Sorry.
Thanks for your question. So actually, we have used we use the ABCD study, which is an ongoing longitudinal study. They have released, I think, there are 2 waves, the baseline and the 2 years follow-up neuroimaging. So we use their baseline, which is like the age of the participants is 9 to 10.
But would it be, I guess, once more of the longitudinal data is released, could you use a similar approach to look at like people who, or not,
Yes.
children who transition
Yes.
to having those mental health disorders?
Yes, we're doing this right now. Yeah, thank you.
Any other questions? I'm not getting any questions on the slide.
I think there was another component to Lynn's question about the validation. So the sample is actually quite large. The total sample is about 11,000 people. So it gives the opportunity to do within sample validation. So you just split the sample, and so you use a part of it as an independent test sample. And completely independent replication in a completely different sample is very difficult because the other samples that are available do not have all the neuroimaging phenotypes that we have used. They have the very basic one. So some sort of partial replication would be possible, but not a complete replication. But splitting the sample within the ABCD is by far the best way of doing that, because you have independent individuals, but a standardized assessment. So you minimize any variation due to the assessment.
Yes, it's just one of the many advantages of having a huge sample size to work with. Margaret, I'm not getting any questions through Slido. I don't know if it's, if you are. Okay. Just a last minute reminder to virtual participants, if you have any questions, to put them in. But were there any other questions from the group here?
See, I had a kind of follow-up question for John Jose. I thought the term about the maternal grandmother was kind of surprising, because it seems like in a cancer workup, you would be asking about family history, especially like a breast cancer or something. Is it, I don't know, is it possible that some centers that tended to stream patients to psychiatrists, for some reason had questions about like the actual word maternal grandmother in there? Or it just seems surprising to me that there would be that relationship.
Yeah, it's a really good point. And the kind of cool thing about neural AI is that it's really seeing things, looking at the whole entire document. And so the maternal grandmother thing was just one component. That wasn't the only reason. I didn't show the entire document. There are some other things that were quite interesting. For example, I think for that patient, the oncologist wrote like, the patient endorses these symptoms and also endorses these symptoms, also endorses these symptoms. And every time there's also endorses like more and more symptoms, the model also thought that was more and more indicative of seeing psychiatry, because we know that, a lot of mental illness, you have more like symptoms and stuff. But so I don't think it was the only reason. I don't think though it was like a specific, like if you have a family history, you go to see a psychiatrist, but things like that, I think is something we need to watch out for in case that's what it's picking up on. But yeah, I think in this case, it was maybe just something that the AI learned was at least slightly predictive.
And any last questions before we wrap up for the morning? Okay, so just a reminder to please go to the polls on Slido and vote for your favorite lightning talk from this first set. And again, I want to thank all of our speakers for doing a fantastic job in presenting their research. So now we do have a break, so please go out. Please do take a look at the posters, enjoy the refreshments, and we're going to ask everyone to come back at 10:30.
So is it of the host? Like, where is the host logged in from?
Sorry?
Where are we logged in from?
No, we don't bother to care about that. We never log into the host.
Oh, okay.
But it's okay though.
Yeah, because I wonder if we can mute like the people, because we keep getting feedback.
less safe for you, it's less safe for them, isn't it? Yeah. You're afraid of. Yep. Yep. Mhm. Right. First one, like, what caused it? What caused the first one? What caused this first attack? With the mask on or just the ball hit him? Yeah. That probably was a significant one. Yeah. So in a way, it's not really a significant injury, this one probably, unless he was really didn't see it coming or something. And he moved his head. So, I mean, in the past, people didn't think much about these injuries at all, right? And everyone survived. And then these people who had a lot of significant concussions, like football, which might some damage, right? So it makes sense that we take care of concussions when there's significant damage, but now there's all this gray area, you know, like what's significant, what's not. So we err on the side of caution now, right? Which then gives the message that you need to self-monitor. So the more you self-monitor, the more you see things too, right? As you know. It's a tough situation to be in. So I think you just gotta kind of say, yeah, you kind of identify what might be the symptoms, give some time, but you don't get catastrophic about it.
Testing. Testing. 1, 2, 3, 4. Thank you. There you go. You put it on yourself then.
Okay.
It's sanitized already. So move that thing a bit down. Close to your mouth.
Okay, let me just.
There, it's
Okay.
in your pocket.
Okay. Maybe I put it down
Flip it
here.
there.
Yeah.
Let me do it for you.
Thank you.
There you go.
Okay, thank
So
you.
let's do it like this. You can
Okay.
do some testing.
I can, yeah, let me take it off. Probably easier when I talk.
Yeah,
Well,
difficult.
okay. Can you hear me? Yeah.
Yes.
Okay,
Yep.
that's fine.
Very good.
Thank you.
So you need to lower your own
Yes, yes. Okay. So Okay. Where did I put my glasses? Okay.
Other side of extra clear. Oh, but really has to be, right? You have to be honest with, if they significantly hit, did it affect losing any consciousness? They twist their neck, or did they just get hit the ball straight on, like happens all the time? It's probably not. So how do you assess that, right? So the idea is you take them out of the game, you talk to them, see who witnessed it, ask them how they feel, and then there you keep them out.
Well, she was herself. How she's kind of developed. There we go. Okay.
Yeah, we gotta use on slide.
Yeah.
Yeah, perfect. We can just minimize it for now. Yeah.
Girls. And it's up to you. Yeah. So these are the masters. To take part in provincial. And but besides that, let's just move on and until he started.
Oh, come on.
More. It didn't last very long.
No.
Their parents were all just straight to the point where they could never get together.
Let me do it.
I think that's mine. That's mine.
That's yours?
USC, this B is mine.
Okay, how about D then? Also yours?
That's mine.
Let me take it out.
You're coaching again with her or not?
Is he going to again?
Okay, they don't use this. Let's use this one.
In the summer?
I'll get some more here. Sorry about that.
No.
Get away.
Okay. Okay.
Here you go. That's all yours.
Okay.
You can copy to your desktop if you like it instead of like this.
Repair. Okay. And maybe I just leave it in there, or yeah.
Yeah, definitely you can keep it in there. And
Okay.
then, remember to remove it afterwards.
At lunchtime, you can come and gather it.
Yeah, I have to leave immediately afterwards because, as I said, I'm actually in a day-long interview panel, and I had to excuse myself because we're getting a new COO, and they asked me last week to join, so there's not much I can do. Sure. Yeah. Okay.
His teacher, was that,
What kind of noise is this?
all of it. And,
Hear something?
I have how much time? Just to make sure. An hour. Okay. So I do leave a little bit or a substantial actually time for discussion, correct?
There should be some,
Yeah.
20 minutes.
Okay.
ideally,
Okay.
or
Okay.
40 minutes of talking and 20 minutes of questions. Does that work?
Okay. Now I need to go back. Okay. Yeah.
Okay, great.
Okay.
Possible to do this work on your own. Possible, what are you missing? Does the extra. Same with all athletes, right? Find a way to get the stuff done.
Okay.
Who's motivated, he'll get it done. Support it. Right. Boys lazy?
They don't like it. Oh, there was I had this feeling that Christine, she did. I thought she was going to make me a birthday party. And I think I told you. And all this. I told her, I asked her, and I said, look, I really don't want to. And then I asked Nathan, thinking he might slip up and jump in. She was. Anyway, she did it. And it turned out that what she did was so wonderful. She said, where she contacted to all my relatives, or a lot of them, and with either people, and asked if they would make a video, and she put them all together, and it was fabulous. And she got some pictures of me from high school, and recording.
Nope, still on.
She did surprise me. Thank God it wasn't.
How did she get you online?
Online, she brought it over.
Oh.
And she brought it over.
Okay.
So, a lot of interesting things. My boss and her two boys and her husband saying happy birthday. Why not? Very high, so they had to. That was really good. And other people did. And she took some pictures too with the company. Past history. I started my first graduate, and I just have a job. And Jeff said, thanks, Mom, for everything. And I thought, wow, that's pretty difficult relationship. So it was really nice. I was really touched. Really touched.
And
Yeah. And I don't look at it. And then what happens if at morning, I'm okay. I have breakfast. And then after breakfast, I start getting the chills and sleepy and just not well. And so I think it's fine.
How do I?
Mute it, mute it.
Spinach tastes really good.
What does? Spinach.
Yeah.
Tastes good.
I, well, first of all, my iron level is up perfectly. I've been having trouble with that. When people check me out with the test. And because I started putting spinach in my scrambled eggs. And I like it so much that will actually wait. And I cook spinach with it and a little bit of cheese. And I like it so much that I mean, and I enjoy it so much I could actually go myself. Yeah.
Who's the Zoom host? Uh-huh. Because I think as a Zoom host, you can mute everybody, right? Hi, Trisha. How are you? Let me see if I can turn this off. Yeah, I'm the next speaker.
You just feel cold?
Is it because well, I think it is because when I was in visiting Dr. I've been complaining about it years ago. When I first got to speak, I said to him, I get chills. My temperature is all the things. I'm either hot, or I've got chills, or I'm warm. And if I get chills, I just put my clothes on, right? But I don't turn the heat up or anything. But these chills come he said he had a little walking out of his office, of his exam room, and his trees and the desk, left desk, and he said, what did you think? He said, they were wearing jackets. They always cool, chilly too. And I thought he was just avoiding telling me that it was a symptom of old age. But I didn't think that you think that I mean, your temperature gets wacky.
Partly true. But it's often related to how much fat you have to insulate yourself, and how active you are, and
Right. I sweat when I
or when you after breakfast. After breakfast, if you go out and get some exercise, you feel better. Challenge it to see what happens.
Right. I'm going towards South Granville Street. South Granville Street today.
Okay. That in?
I think it's Julie. And I'm doing 6 times. That's what she said. People, when after she hired that lady, my health, Justin come back. Yeah. And
Do you have the keypad?
I made sure I said,
Hi, Tim.
Embarrassing for somebody.
So it's on.
No.
Right, that's what happened.
Oh, there. Yeah.
Temperature. It's getting cold outside. Cold.
Working now? Yeah.
Two degrees higher than we ever have, so it's not cold.
Hey, anyone in the meeting, can you mute yourself? Please mute your microphone. I can hear someone talking. Can you mute your microphone? Please mute your microphone.
Put water. You put water in it.
Oh, yeah.
You gotta put cold water. Blows air over the water. But for short.
Hello? Please mute your microphone. We don't know.
So I told her, you're in charge it for an hour. And so important. And
Can we assign it to, are you like?
Water. Does. It does, does it? I'm asking you.
I don't just
Oh.
because we sometimes have
I thought it maybe this.
Okay, they said it was on the.
a sign at the door which tells them how to hold us.
Right.
And so Chrissy said we'll send it to my house because her husband's working, but we'll put it out. And so just while I had to talk.
The lightning talks are loaded in a separate file after yours?
I didn't load mine down. It's still on the USB. So I'll just grab the USB afterwards and then it should be on there.
Okay. And you have 56 slides.
Yeah, but I'm not that. I always have, but there's some extra at the end. Yeah.
Right.
Yeah. And I don't appropriate place in the window because right in the I don't think they know.
Right.
Because nobody else has it.
Well, how would that cost?
Yeah, I'm comfortable.
Well, how much? How much? Is,
So you don't have to do anything because you will be on.
Okay. That's why I'm not wearing a mask.
You don't need to use the mask. You cannot use the mask already. You cannot hear you. That's why we mumble.
Sorry, so we want to disconnect? I can't mute all? Okay, so what do I need to do? How do we do that?
You don't hear any noise.
Okay, I'm gonna. Sorry. When I hit mute all, it muted the lecture, so it has to reconnect that lecture. Sorry. How long do we need? He needs a few more minutes to log in the room again because I accidentally muted it.
Oh.
Yeah. So we're not supposed to mute all. Okay. Yeah.
We're working on it.
How long will it take? Because you don't have it. How long will it take?
Now my slides are gone.
Okay, I was told that we are good to go at the moment. And so it's my great pleasure to introduce Dr. Christian Schutz, who is professor at the UBC Department of Psychiatry and who will be giving the plenary talk for this presentation.
Go ahead.
So Dr. Schutz has been an academic faculty member at UBC Psychiatry Department since 2009. He's also a research scientist at the Center for Health Evaluation and Outcome Sciences and the Research and Education Medical Manager at Burnaby Center for Mental Health and Addiction. He leads the Brain Lab, where his research team investigates the psychoneurobiological mechanisms underlying cognitive, affective, and stress-associated aspects of decision-making and impulsivity, specifically as they relate to substance use disorders and concurrent mental disorders. His work has been recognized by various notable awards, including a NARSAT Independent Investigator Award and the UBC Faculty of Medicine Outstanding Academic Award in 2018. We are very pleased that he will be addressing a topic of addiction, specifically addiction as a disorder of decision-making, which is a topic that has both scientific, mental health, but also wide societal ramifications. Dr. Schultz.
Thank you very much, Sophia. Thank you for the introduction. We did have a little bit of technology problem. Shows how addicted we are becoming to technology, that we are not able to start until things are sorted out. But I think we had the acknowledgement this morning. I do have a number of grants and I am on advisory boards, but that is not conflicting with my current presentation. What I want to discuss today is first talk a little bit about the addictive processes, then present some of the work related to that in my brain lab, and give a summary and then have the possibility for questions. I'll try to make it a little bit faster and see that we have enough time at the end to discuss. When I talk about addiction, I'm really focusing on substances of abuse. And I think that the discussion around substance of abuse disorders and addiction are sometimes confusing and confused. And what I want to start out with, discuss it a little bit. One thing is that if you take substances, you can have an intoxication and you can have an addiction. These are two separate things, but they're somehow interacting. But I think often we are confused on how these two aspects of substance use are interacting. I want to do an example. The idea often is that addiction is driven by the intoxication and that people are seeking the intoxication as the driver of addiction. I have two examples of two substances here. One is psilocybin. With psilocybin, we know that people have a severe intoxication, but there is no risk of addiction whatsoever. So despite this being a substance with a severe, so to say, intoxicating effect, no risk of addiction. On the other side, tobacco. With tobacco, we don't have the traditional intoxication, but we also know that this is a highly addictive substance. So while there is some interaction between intoxication and addiction, it is not as direct and as not closely linked as a lot of people think. Berich, for example, tried to make a differentiation between liking, which is driving us to use the drugs initially, and then wanting, which is actually driving the addiction. While there is a relationship between the two, they are not as closely related as people generally tend to think. So what is addiction? If we talk about addictive processes with substances, obviously you have to start using the substances, otherwise you cannot become addicted to it. And it's generally not within the first few uses, but it takes a lot of training and experiences of drugs to develop addiction. Most people who use drugs don't get addicted to it. Even in substances which are highly addictive, like tobacco or nicotine, only 1 out of 4 people who try tobacco really kind of come to the level of that you would say they are addicted to the substance or they have a nicotine use disorder. So even if you are using the substance, you will stop in between, and people, so to say, the stops in between can be shorter or longer. But once you have developed an addiction, you are at high risk of showing the addictive behavior if you start using the substance again. And what is addictive behavior? It's excessive drug use. It's also a narrowing of behaviors. It's something called loss of control, and then craving and relapse. Interestingly enough, DSM only introduced craving as a symptom of addiction in DSM-5. Before, craving was not a symptom of addiction, even though all the clinicians used it all the time. The reason being that actually craving is related to relapse, but not as closely related as most clinicians think. When people talk about substance use disorder and addiction, they often think about also withdrawal and tolerance, which are the physical symptoms. Actually, the physical symptoms are not necessary to diagnose a substance use disorder. What we see in a lot of substances and substance use is that the initial high or the intoxicating effect actually become less and less over time. And withdrawal symptoms and changes in the wanting behavior is what is actually driving the substance use disorder. In a slide which gives a definition of what addiction is, one thing discussed was the feeling of loss of control. And I think that is something which is quite central to our understanding of addiction, but it is surprisingly enough not a driver for people to go to treatment. There is a big difference between the concept of loss of control subjectively and objectively. When people come to treatment, it's generally the family, it's the work, it's friends who are indicating that the person is having a loss of control over the substance use. When you talk to the person, there is no feeling of loss of control over the substance use. Person can step outside of their subjective experience and look at their life from the outside, and then they come to the conclusion, yes, it looks like I'm losing control. That is what you do with motivational interviewing. You're actually trying to get an individual to take an outside look and replace this, so to say, subjective lack of loss of feeling of control by taking the outside look and seeing that, okay, yes, I have a divorce, I lost my job, I lost my driver's license, and it all has to do with alcohol. So even though I feel that's not the alcohol, but there seems to be this relationship. When you develop abstinence, or when you are using drugs, there are two aspects which are kind of driving relapse. And one is cues and one is stress. So I will talk a little bit about cues and stress, as they are the drivers of relapse and of approach behavior. But first, again, to the original kind of ideas regarding substance use disorder. You start out with recreational sporadic use, then the use becomes more intensified, and then actually we have this aspect which is called loss of control. But important is also that with kind of like increasing use of the substance, it also becomes a bigger part and a higher priority in the patient's life or in the person's life. Most users are actually not patients. And there is less interest in other rewarding activities. There is less interest in working on function in the family or in occupational. And the substance use takes over a wider space in the life. So again, there is this stepwise progress from pleasurable use to it becomes more desirable, to it becoming a need, and then it becoming a compulsion. So there's often, you know, when we do categorical diagnosis, we have to make a point where we say, okay, this is substance use disorder, this is not substance use disorder, but this is a process which is a stepwise process. I always say that if you have to take active breaks from using substances, like saying, okay, I'm not going to use in the next 5 weeks or so, this is an indication that you might be on the first steps towards substance use disorders. It doesn't mean you have a substance use disorder, but it's a step towards it. Substance use disorder have a huge impact on people's life. This is a study which I was quite impressed with, because what we see in the more severe forms of substance use disorder is that individuals develop also some impairment in decision making. This is the IOR gambling task, where people have to kind of make decisions based on information they have, and people seem to make worse decisions when they're in an active state of addiction. They are more impulsive, they are more easily stressed out, they are more chasing, so to say, the big events. And what you see here is actually an improvement in individuals who abstain from the substance over 24 months. So within at least the first year, you generally see stepwise improvements in people's decision-making, in people's overall life. So what it means is actually that substance use disorder has severe impact, and that the impact is long-term, and that actually expecting from somebody who is struggling with severe substance use disorder to be effectively treated within 4 weeks is unrealistic. There are different ways of looking at substance use disorder and addiction. One aspect which I like to emphasize, and it's more a attempt to try to understand it from a clinical point of view, is to look at dual process theory. So there are a lot of dual process theories. Kahneman, who also was working here, for example, has written a book about thinking slow and thinking fast, which is also along the dual process theory, but it has been invented and reinvented over and over again. It's very intuitive. It's probably not completely consistent with, so to say, the neurobiology and the neuroscience, but it gives you a first idea of what might be going on from a phenomenological point of view. So there is an understanding that we have more top-down processes, which are more planning processes, and bottom-up processes, which is what is driving us. So what are our urges? And both aspects are very important in decision-making, and they're important in decision-making differently in different contexts. Voluntary thinking and voluntary planning is generally when there are no immediate demands to you, then there are no stress, there are no competing kind of like situational things you have to respond to, while the habit-based, skill-based automatic responses are more responses which are important when you have something where you need to respond immediately. And what we can see is that the interaction between more voluntary or more top-down processes and the bottom-up processes can change quite substantially. One aspect which I think is very important is that we all think ourselves of as conscious persons, and we have the feeling that we make conscious decision-making. And we are aware that actually, probably a lot of our decision-making is way less conscious than we think it is. And that we also kind of like sometimes come up with conscious explanation for decision-making, which we have not done consciously. That is something I always discuss with individuals who sometimes come up when they explain their substance use with explanations which don't make sense. And when you start discussing with them, they also realize that they don't make sense, but they need to kind of like come up with an explanation why they, as an intelligent person who is in control of their life, make decision which doesn't seem to be consistent with the self-perceived conscious processes. As I said, there are many different ways of looking at it. Sorry, this is flipped around. So the bottom-up parts on the left and the top-down on the right. So it's the reverse. But obviously, the controlled processes are more the prefrontal processes. They are more the conscious processes. They are very slow. If we do plans, we have to take a lot into consideration. And so these are not the processes we need if we have to make a fast decision-making in the situation. And they seem to be way more controllable, while the question is how controllable they are, because you at the same time have the automatic processes, which are less conscious, which are quite efficient, quite fast, but they are kind of more triggered by the immediate surrounding environment. So when we work on understanding substance use disorder and treating substance use disorder, you can focus on the aspects which are driving relapses. And we are focusing on impulsivity, which means to make the more automatic, the more kind of guts decision and less the planned decision. We focus on stress. Stress is typically a way of inducing a relapse. People under stress are more likely to use substances. And cues, which are environmental factors which trigger approach behavior. Let's start with impulsivity. This is an overview and very similar. The top-down processes are more the prefrontal cortex, the dorsolateral prefrontal cortex, mainly the right inferior frontal gyrus and the ventromedial prefrontal cortex. These are three areas which seem to be important to process the top-down approaches. On the other side, in addiction, we focus on the nucleus accumbens and the amygdala, which kind of are involved with salience, making sense or importance of a cue or environmental trigger. Then the amygdala probably is more with the valence, which kind of indicates if you're need to avoid or approach a situation. And these are all related to the motivation, which again is very closely related to decision making. One way of altering these processes may be with TMS. And I know Fidel will present in the afternoon on TMS, and we are doing a study with him. Most TMS approaches have been targeting the dorsolateral prefrontal cortex. For example, the approved treatment of depression is based on the dorsolateral prefrontal cortex. And a lot of groups have focused on the ventromedial prefrontal cortex as a way to change OCD and impulsive behavior and to allow individuals to have stronger inhibition. We have been focusing on the right inferior frontal gyrus because there's good evidence that it might improve inhibition. This is work done by Carling, who has also a poster. She has a lightning talk. Okay. The second aspect is stress, which is more unspecific. Here, Karina, who is having a poster on a very different topic, is studying the impact of THC and CBD on stress and looking at how these THC and CBD impact on stress using fMRI. There is always the report that individuals take THC and CBT to reduce the stress response, but this is not very clear if that does really kind of like have an impact on the stress response and what kind of impact it does have. And finally, on cues, environmental cues are very important in triggering people to start behavior. And there have been a lot of work on trying to understand craving. And so you can differentiate between unspecific craving, background craving, which has regularly been measured, but you can also do cue-induced craving. And together with Olaf Krigelsen from UVic, we developed an app and portable EEG, where we can measure the P3 of drug cues induced evoked potentials. And you can quantify the evoked potentials, the P300, and they give you a measure of the stimulus evaluation. So if the individual responds stronger to the cues, it is more likely that it has an impact on decision making, which in the form of cue is approach behavior. So this is kind of like has been around in the discussion a lot. Using a portable kind of like EEG and using this app where you can provide triggers in the form of cues will help to not only have, so to say, the subjective craving, but also compare it to an objective response, which doesn't have to be conscious. And what we are also looking at specifically is incubated craving. So from animal work, we know that specifically with stimulants, craving in cue-induced craving initially does not go down, but it goes up. And previous data show it might be going up for 2 to 3 months. So that would be very important if we could show that, so to say, actually the trigger has a stronger effect on the individuals in the first 2 to 3 months than the currently accepted thinking that it's actually kind of going down after the initial abstinence. So that after 2 or 3 months, people are generally already kind of discharged. And that might be the point where the craving or the trigger-induced approach behaving is the highest. This is actually the data, not our data. This is a study done by Rita Goldberg in New York. And we have been working with her on developing our measurement. This is a study which is a reanalysis of some data which we did a while ago, looking at both cues and stress and predicting relapses in smokers. We followed up smokers for 6 months, and before they started their treatment, we said what predicted a relapse, and we found very different areas in the brain to be associated with relapse. For cues, it was actually the attention areas like the dorsolateral prefrontal cortex, parietal, then it was anterior cingulate and premotor cortex. And these are action areas which are associated with attention, with action initiation and motor activation. So it means that those who had a higher response in the brain to get activated to approach behavior, I would say, or at least to some action, were more likely to relapse. With stress, which was forced arithmetic, where we did have a person kind of talking to the client in the scanner and asking them to be more kind of like involved in the arithmetic and say, can you please try to give an effort? So because if you're not doing an effort, then we cannot use the data. So we put stress on the individual, and so that actually led to the result that those who, that the insula was the main area which was predictive for relapse. We did not really have, so to say, the differentiation if the relapse was induced by stress or by cues, but we know that it's different pathways which are leading to these separate aspects of relapse. What I've been talking about mainly is so far the disorder and mechanism underlying the disorder, but we also can look at risk factors and the consequences of disorders. And generally, when you look at risk factors, it's the prevention work where you attack or approach or work with risk factors and trying individuals to prevent from developing the disease. Then you have the treatment, so to say, if somebody has a disease, you try to help the person to overcome the disease or deal with the disease. And finally, it's often if you cannot change the disease, you try to work with the consequences. Yeah, sorry. Okay. No, sorry. I took out 1 or 2 slides. That's why. Okay. As I said, the risk factors is mainly public health, the disease is mainly medicine area, and the consequences is mainly the harm reduction approach, which is public health, medicine, and also law enforcement, for example, because the consequences can not only be like for the individual, it can also be for society. What I have been discussing is about treatment. We also have looked at the mechanism, but there's also the question of infrastructure. We are also doing some work with infrastructure. This is a study we recently published looking at the emergency room use in BC. And what you can see here is that these are data from 2007 to 2017. So that's before COVID. And this is work done together with Ruth Laverne from SFU. And it's based on BC POP data. So we get all the emergency room use and look, what are people coming for to the emergency room? And what we can see is that actually the traditional, so to say, psychiatric disorder stayed pretty similar. And substance use, to most people's not big surprise, is the one which has been increasing massively. And I actually assume that it's still increasing. The second is, to our surprise, to some degree, it's anxiety disorders were also kind of like increasing over time. One possibility is that anxiety might be also associated with substance use. A lot of people who have some form of limited withdrawal or kind of like problems with substances are also identifying these withdrawal symptoms as anxiety. So it might be related, but we don't have any proof, and it could be something completely different. Before kind of like going over the questions, I also want to at least touch 1 or 2 aspects of the overdose crisis, simply because that is probably on a lot of people's mind. And different substances obviously have a different ratio between the use, be it recreational use, and the fatal use. And when you look at it, Here it's like, you have marijuana, LSD, psilocybin. They are very safe drugs. It's almost impossible to kill yourself with these drugs, if it's possible at all. And then on the other hand, you have heroin, where the dose between, so to say, the intoxication dose and the fatal or lethal dose is 5 times. With alcohol, it's about, where is it? Here, 10 times. So these are data which are thrown around, and they vary a little bit. If they're based on animal studies, they're a little bit different, and also kind of like the data are often not validated that well. And there are some other aspects to it, which I will talk about while showing these slides, because it's not only the relationship of the substances, but also the amount of the substances. Here, my preferred substance of abuse, caffeine, very safe. I mean, if you look at it, it's very hard to accidentally take the right amount. You could buy it as a substance online, and kids have overdosed on caffeine by using the actual caffeine substance. There are medication we know have a certain risk, but fairly safe compared to the substance of abuse. Here's cocaine, and already one thing you can see is we in the province have been moving from a lot of people using cocaine to a lot of people using methamphetamine. You just need way less substance to get the intoxicating effect, and you also need way less substance, just absolute amount in overdosing. And this is a tendency we have seen both with the stimulants and with the opiates, that going away from plant-derived substances, which are very expensive to produce, need to be transported, and then are used here to industrialized production of methamphetamine and fentanyl locally, and you need small amounts. This is, now we are getting to heroin and fentanyl. And when you look at the amount, this is, by the way, these bags are 2 centimeters. They're really small bags. And the numbers there, they vary a little bit because they are numbers taken from different sources. And one thing which is very important is that I talked about in the beginning about tolerance. So people develop tolerance. And for example, methadone, if you look at it, the lethal dose is about 50 to 75 milligrams, according to this source. That is the standard treatment dose. So I have patients which are way beyond 200 milligrams. So the patient is taking a dose which is high enough to kill 3 persons. So yes, we are talking about toxic substances. It's the substances which are toxic. There's nothing additional toxic to the substance. It's all of these substances are highly toxic. And when you can see heroin, there's at least a little bit you can see. With fentanyl, actually, it's so effective that how much do you think, how many grams, kilograms, tons do you think you would need for the whole province to have a fatal overdose? It's about 5 million in the province, a little bit more. So with 1 kilogram of fentanyl, that's enough to kill about 500,000 people. So that means about 10 kilograms of this substance is enough to give everybody in the province a fatal overdose. So that's a very, very effective substance. And dealing with it, it's not only you have this variation in the small window between effective use and fatal use, but you also are dealing with on the street with kind of like really minute amounts. One aspect is also we are doing a lot of substitution treatment, and that is considered to be standard. But the quality of the substitution treatment is very important. When you look at here, this is, so to say, the risk of overdose and fatal overdose when off treatment. This is when people are on treatment, but during the induction phase, you have an increase of the risk. And if you get off treatment, you also have an increase of risk. So if you have people on it for a long time, it's very safe. But if you have people going on and off, like low quality substitution treatment, you're actually increasing the risk of fatal overdose. These are aspects which I see are very little discussed, because it's not only important to provide, so to say, substitution therapy, but we also need to discuss the quality of that. And that is one thing we have a lot of substitution treatment where people go in and out of the treatment constantly. So actually, you're not helping the individual. Before summarizing, and I try to be a little bit faster because of the initial time lag, we are also kind of doing digital care, which we were working more on. Miriam has been doing that. We have found, for example, very interesting results using wearables. Our patients, on average, sleep about 10 hours at the center, and that means on average 10 hours. And that center is the Redfish Healing Center for Mental Health and Addiction, where people have both substance use disorder and mental disorder. And they are sleeping 10 hours, and they're actually constantly asking for more sleep medication. So there's an interesting process going on, which a lot of people have not, even in our setting, realized that people are sleeping to that degree. And the sleep is actually maybe less effective. It's not that interrupted according to the wearables, but that's something we are looking more into. Then we have started to do psychedelic assisted therapy, which we will kind of like make more of a focus of our work. And then we are working also to develop the patient partner track so that we have more interaction with our patient partners. And we have started this process a year ago, and I think that's a very important process, that we get our patient partners involved in all of the studies. So in summary, addiction is a complex complex behavioral changes. They have an impact on motivation and decision making, and they are long lasting. The loss of control is an appraisal of their behavior, not so much as a subjective experience. Drug seeking in individuals struggling with addiction is initiated generally outside of consciousness, which is often not appreciated. And most people with addiction who present for help have other psychiatric problems as well. That is one example of the major focus of my work, which I have not touched because it would have been too much. Thank you very much.
This was fantastic. Thank you very much. It's sort of powerhouse of information about neurobiology, treatment innovation. Fantastic. I'm sure there are many questions in the audience, and I definitely have a few already from our virtual attendees. So if you want to say something, just wave. Yes.
Dennis, your voice is loud enough.
Great presentation. Thank you very much. So I'm wondering, in terms of substitution treatment that you're using, now that we're seeing like more laced fentanyl in the Canadian market, would you say that substitution treatment has changed, given that there's like different substances in the supply, and with that happen?
Yes, I think we are almost in a Cold War situation, that the dealers are beefing up what they are providing, while treatment are beefing up what they are providing. What it means is, it used to be like methadone was the substitution which was early applied and which had a long term and was given regularly. Now we are trying to kind of like use other substances to pull in more patients into treatment. The original European model was actually to get nearly everybody into treatment. And that was actually also why heroin was provided as a substitution treatment in Switzerland. The idea was we get only to 60 to 70% of the patients into the treatment. And so how do we get 80, 90% of the patients or the users into treatment, or the addictive users? And that is the original idea. Here we have started providing these additional substances while we have a level of about 30% of the users in treatment. So it's a very different situation. Now, also with fentanyl, we have this situation that the substances are, so to say, stronger. We have more tolerance development. People are using way more substances, and the treatment becomes more difficult because if individuals have developed these tolerance and you try to work with them, now it takes much longer time to get individuals on lower doses or safer doses and on even to try to get them off. So we have now individuals which have methadone plus Cadian, which is the slow-release morphine, plus fentanyl. And that in itself is a problem, that we have 3 different substances in the substitution. And each of the substance is given at a level which is lethal for a person which is not using the drugs. So this is a situation which is risky also with sharing drugs. That is, for example, we have people with patches, you know, if they give patches to somebody who is not tolerant. So that is a very difficult situation. And we have also persons which come to us and say, no, I don't want any fentanyl anymore. I'm not getting any high from fentanyl. I want carfentanil. And so they're actively seeking, so to say, they're chasing this high, which are not getting, and developing more tolerance. And that makes the situation quite difficult. I think there is also the attempt, so to say, to replace the treatment to have people come buy on the illegal market. But the problem is that you have now a competitive situation. And another thing which we are seeing now in the market is that the drugs are cut with benzodiazepines. So the problem is when we kind of now use naloxone, we have individuals who get high doses of naloxone, and it's not effective because they have also high doses of benzodiazepine. So I think the idea of getting everybody into treatment is very well, but in BC the epidemic is rampant. You know, we have so many individuals. Like often, sorry, keep on talking, but there is also the Portugal model. In Portugal has 5 million people, and they were talking about the crisis when they had 100 overdose deaths, and we are talking here now to close to 2,000. So it's a very different dimension.
Christian, we have about 4 more minutes. I know.
I try to be short.
Very short. Yes. Very. I mean, what you're saying is terribly useful and interesting. Difficult to interrupt. But the next question is basic, and it comes from virtual attendee Mohammad Ali Niku. I say, how do we measure the addictive potential of different substances?
Yes. Well, the traditional way of measuring it is with animal work and self-administration. If a substance is self-administered by animals, then we assume that it has similar effects in humans. There is other ways, condition, place, preference to do it. So there obviously is also to keep in mind that there are huge variation in humans, because most individuals who use substances are not addicted. It's always like 10, 20, 30% the most, which become addicted. So the majority of users is actually not addicted. And we know also that 20% of the substance users use about 80% of the substances, or even 10 to 90. So there's a large group of users which use limited, and then there's a small group which use a lot. And among the group who use a lot is probably the ones who are addicted.
And our final question that comes from Trisha.
Yeah, thank you for that fantastic talk. I was really interested by that top-down versus bottom-up processing, and I was getting the impression it's probably easier to target some of those top-down processes in interventions. I was wondering if that was your impression as well, and whether there are any interventions that are kind of targeting more of those bottom-up unconscious processes as well.
Very nice question. Thank you, Trisha. That's one of the big problems we are having. Most of our therapies are actually top-down therapies. CBT, psychotherapy, you have to understand, you have to kind of learn. And that is something a lot of people we are dealing with have problems with. So the more capacity you have cognitively, the better the top-down processes works. We are actually working on trying to develop bottom-up processes. And bottom-up processes are closer to relearning action skills like binding a shoe. Well, probably nowadays nobody knows what that is, but you used to have laces, which you need to learn to kind of do, and that you do over and over and over again till you know how to do it. And we are testing out an approach at the moment where individuals get cues and then have to push them away, and they train that. And people don't even need to know what they're doing to kind of have an effect from that. It's the automatization process and the retraining process. And I see a huge opportunity actually with digital health, because computers are good in doing the same thing over and over and over again and presenting it to you over and over again. And so all forms of retraining or on automatization, different processes where you do things and you can get rewards for doing that. So because people do these things with gaming all the time. So yes, we have to kind of work on developing more tools from bottom up, and there are possibilities.
Fantastic. A great way to end our plenary talk and the discussion. Thank you all for participating, and thank you, Christian.
Thank you.
This is still yours?
I have it here as a. No, I had more.
So give us a minute to move into the next session of the lightning talks, and I am giving the baton to.
Thank you, Sophia. Yeah. Is it turn mic audio sources? When she comes up, we'll need to activate that. Yeah. It's on. Okay. It's a pleasure to introduce and moderate the second set of lightning talks. And unfortunately, we have one speaker wasn't able to make it because of illness, Angela Rusillo. So we're going to start with Judy Chang from the Raymond Lab. And she's a master's in neuroscience student. And she's going to be telling us about cognitive and motor deficits in HD Huntington's disease model mice. So let's see. Judy.
Hi, everyone. Can you hear me okay? Okay, so I'd like to first thank the organizers for allowing me to speak today. And my project involves looking at age-specific motor and cognitive deficits in the Q175 mouse model of Huntington's disease. So to start, Huntington's disease is a neurodegenerative, Huntington's disease is a neurodegenerative disorder caused by a CAG repeat expansion mutation on the Huntington gene. leading to symptoms such as loss of motor control and involuntary movement, as well as cognitive deficits and psychiatric disturbances or mood changes. And the brain region most deteriorated in Huntington's is the striatum, which is important for motor learning and motivation. So we used the Q175-BLACK6 knock-in mouse model of Huntington's, which basically has the same mutation in the mouse genome as that found in human Huntington's patients. So previous studies have found motor deficits starting at around 10 months of age in this model. And we were interested in looking at more sensitive measures of behavior in this model using commonly used mouse behavior tasks. So one example is combining the deep lab cut tracking software with the new B-side behavior clustering algorithm to look at specific behaviors in the open field. And we had both males and females in our experiments at both pre-symptomatic and symptomatic disease stages. So this is the open field task. You can see the mouse moving around in an open chamber, and we just leave them in there for an hour and look at its natural behaviors. And the labels in the video are generated from deep lab cut, while the bottom graph shows the visual representation of the clusters extracted from the B-side program showing different behaviors. Next, for the accelerating rotorod, we place the mouse on a spinning rod and see how it learns to stay on the rod, and the standard measure would be to look at its latency to fall. But we were interested in looking at more specifically how the mouse is learning to stay on the rod. So we developed a new foot slip analysis that basically measures every time the hind paws of the mouse falls below the bottom of the rotorod. And lastly, for the water T-maze task, we place a hidden platform in either the left or right arms of the maze and see how quickly the mouse is able to find the hidden platform once released from the starting arm. So that would be for the training phase, which measures motor learning and memory ability, whereas in the reversal phase, we place the platform in the opposite arm and see how quickly and consistently the mouse can relearn its new location, so overwriting its existing memories of where the platform used to be. And that would be a measure of cognitive flexibility. So showing just some of our data here, in the older male group, we saw some genotype differences in the behaviors extracted from B-side. For example, the Q175 animals showed more turning left and rearing behaviors, while the wild types showed more grooming. And this is interesting because when you place a mouse in a new environment, they have a natural tendency to engage in more stereotypical type behaviors the longer they stay in a new environment. So that's what we see for the grooming type behaviors in wild type, while we don't see this pattern in the Q175s. We also saw more foot slips in the Q175 animals, which was expected. And comparing with the older female data, some behaviors were consistent with the males, such as turning left, whereas in other behaviors like rearing, we didn't see the same genotype differences in our female group. Also, there was an interaction effect in the foot slip data where the older females only show a deficit later on in training and not in the earlier stages of rotorod training. So there do seem to be sex differences in this mouse model. And lastly, for our younger males, while they had no trouble in learning the task in the training phase, they had problems with relearning its new location. And so it seems like the younger males may be showing deficits in cognitive flexibility at the younger age. So these experiments tell us new information about the sex and age differences in Q175 animals. And moving forward, the next steps would be to look at brain activity during these behavioral tasks to try and identify which signaling mechanisms are altered in Huntington's and how we can try and restore motor learning ability by manipulating these underlying brain circuits. Thank you for your attention.
Thank you, Judy. I just want to remind people that we're going to hold our questions until all speakers have presented. And our next speaker is Priya Yoha. And she's going to speak about childhood maltreatment and relapse. Let's see. and she's a member of Dr. Christian Schutz's group. Let's see, she's an undergraduate who just graduated. Congratulations this May.
Thank you. Hello, everyone. I'll be presenting my undergraduate thesis, which looked at the relationship between childhood maltreatment and relapse in patients who have both a mental health disorder and a substance use disorder, or concurrent disorders for short. So over 250,000 Canadians currently have concurrent disorders. Now, people with concurrent disorders have higher rates of relapse, overdose, and rehospitalizations compared to other mental health populations. These repeated overdoses and rehospitalizations as a result of relapse is contributing to a substantial portion of the $11 billion burden that substance use currently has on our healthcare system. So identifying risk factors for relapse in the clinical group most vulnerable for relapse is pretty important. Reported by over 60% of concurrent disorder patients is childhood maltreatment or childhood trauma, which may be one risk factor for relapse. So in this study, 210 inpatients at the Redfish Healing Center for Mental Health and Addiction completed the childhood trauma questionnaire, which assesses physical, emotional, and sexual abuse and physical and emotional neglect experienced before the age of 18. Now, to give you a brief overview of the sample, patients averaged 36 years old, 67% of them were men, and the majority of them were white. About 60% had a disorder on the psychotic spectrum, and 70% had multiple co-occurring substance use disorders, with stimulant use disorder being the most common. And the reason why these pie charts don't add up to 100% is because many patients had multiple co-occurring substance use and mental health diagnoses. Now, relapse was defined as a positive result on an alcohol breathalyzer or a urine drug screen test for amphetamines, cocaine, opioids, and cannabis. So in this study, 36% of the patients relapsed at least once during treatment, and about 40% had moderate to severe levels of childhood maltreatment across the subtypes of abuse and neglect. Now, for the actual results, higher childhood emotional abuse, sexual abuse, and physical abuse predicted a greater likelihood of relapse to alcohol while controlling for the length of stay in treatment, average alcohol use before treatment, as well as the tendency to minimize and deny childhood maltreatment. So this graph here is showing the relationship between the probability of relapse to alcohol and emotional abuse scores, but the graphs looks very similar for physical and sexual abuse scores as well. Here we can see that for a 10-unit increase in emotional abuse scores, so going from having a low abuse severity of around 5 to a moderate level of abuse around 15, there's a 12% increase in the likelihood of alcohol relapse. Interestingly, there were no effects observed for physical or emotional neglect in alcohol relapse, and there were no relationships between the forms of childhood maltreatment and relapse to the other substances that I mentioned. So what we can conclude is that childhood abuse, but not neglect, is a risk factor for alcohol relapse during treatment for concurrent disorders. Now, since this is the first study to link childhood abuse to relapse in this clinical population, more research is needed to support my findings before any large-scale changes to treatment programs or clinical practice can be made. Thank you all for listening.
Thank you, Priya. And our next speaker is Carly Luciani. And Carling is a master's neuroscience student in the Schutz lab. Let's see, and is also going to be speaking about substance abuse. And childhood neglect again.
Okay, hello everyone. My name is Carling. I'm a master's student here at UBC studying neuroscience, and I am in the brain lab of Dr. Christian Schutz. And just a little disclaimer, this is the same data set that Priya used in her previous presentation. So I will be presenting on how childhood neglect predicts perceived quality of care in patients with co-occurring substance use and mental disorders. To start off with a bit of an introduction with background information, past studies have found a high prevalence of childhood trauma among those patients with co-occurring substance use and mental disorders. Also, childhood trauma is associated with long-term mental health complications. But even with this knowledge, potential patient treatment challenges have not been investigated. Also, there is a higher perceived quality of care and treatment satisfaction consistent with improved outcomes in these patient treatment centers, which shows the reliability of a patient satisfaction measure. Which brings us to the aim of this study, which is to investigate the relationship between childhood trauma and perceived quality of care in patients undergoing drug use and mental health treatment. So there was adult patients aged 19 and over in tertiary care for co-occurring mental health and substance use disorders, recruited following 1 month of admission into the treatment center. The patients also had to be stable on medication to participate. The participants underwent one single session comprising of self-report questionnaires, and there were 2 questionnaires that I was specifically interested in. So the first questionnaire is the childhood trauma questionnaire, which assesses 5 types of childhood trauma. There are 2 types of neglect, which is emotional and physical neglect. And this is defined as a lack of action or failure to provide emotional or physical needs. There are also 3 types of abuse: emotional, physical, and sexual abuse. And this is when the child is at risk of harm, emotionally, physically, or sexually. The second measure that I was interested in is the inpatient consumer survey, which assesses a patient's perceived quality of care at the treatment center in 6 domains. The first domain is outcome of care. So if the patient is able to effectively deal with their illness following treatment. There is dignity. So if the patient feels that they are respected at the center and are treated with dignity. There is rights. If the patient feels that they are able to express disapproval with the treatment strategies with the healthcare providers. Engagement, if the patient feels that they are involved in their treatment process. Environment, if they feel that their environment at the treatment center is safe and inclusive. And lastly, empowerment, if the patient feels that they are empowered and knowledgeable about their illness. From our results, we found that of the 259 inpatients who completed these surveys, 66.3% of them had a mental disorder in the psychotic spectrum. 35.1% had a mood disorder. 30% had some type of anxiety disorder, and 73% had more than one substance use disorder. We also did binary logistic regressions to see the relationship between childhood trauma and the inpatient consumer survey. We added the minimization denial subscale from the childhood trauma questionnaire as a covariate to make sure that we are making sure that we are not underreporting traumatic experiences. So from this first graph here, we can see that emotional neglect independently predicted poor patient ratings on all 6 of the inpatient consumer survey subscales: outcome of care, dignity, rights, empowerment, engagement, and environment. We also found that physical neglect independently predicted poorer ratings on the inpatient consumer survey for outcome of care and dignity. However, there was no relationship between childhood abuse and the inpatient consumer satisfaction measure. This leads us to a few conclusions. First, that it was childhood neglect, but not childhood abuse, that was associated with a greater likelihood of having these negative perceptions about quality of care. This is also the first data set to suggest that history of childhood neglect is linked to potential treatment challenges in patients with co-occurring mental health and substance use disorders. And finally, further investigation is needed to assess past childhood neglect and quality of care as they relate to outcomes in treatment, so we can better understand how trauma-informed care can be incorporated into these treatment centers. And this concludes my presentation. Thank you.
Okay, thank you, Carly. Our next speaker is Angel Wang. And she's a research coordinator with Daniel Vigo, and is going to be telling us about e-mental health and student surveys. Let's see. Do you want to? Yeah.
Because there's two.
Oh. That's the right place.
Can everyone hear me? Great. So yes, I will be talking about the Student E-Mental Health Survey and some specific findings from that survey. So to provide a little bit of context, this is a 4-year multi-site study for Canadian university students. And so we actually, our 4 sites were active at UBC, SFU, McMaster, and the University of Toronto. And so we're actually coming up on around 30,000 participants across those 4 sites now. And it is in collaboration with the WHO World Mental Health International College Student Initiative based at Harvard Medical School. And so we've adapted their survey for Canadian context. And so let me tell you a little bit about the logistics. Just so you know here, from now on in this presentation, I'm only going to focus on kind of UBC specific data, but throughout the kind of different 4 sites, we all use the same protocol and processes. So what kind of happens is this is a randomized stratified sample of 350 students are polled at each campus, and they're invited each week to participate in this survey. They have around 10 days to respond with 2 reminders throughout. And after those 10 days, the initial survey closes, and we actually pull a randomized sample of 70 non-responders. So what I mean by that is individuals who have not engaged with the survey at all, and we do a kind of follow-up with them through both phone and email, a personalized email from the PI for those who don't have a phone listed. And so what I want to kind of draw your attention to here is kind of how that A is decreasing the non-responder bias amongst the sample, but also increasing our overall response rate. We can see that for the kind of easy to reach individuals who respond to that initial invite, around 27% of them actually respond. However, when we then account for the kind of follow-up with the non-responder population, our weighted response rate actually increases to almost 40%, which is quite noticeable. And so through the survey, what I want to highlight a couple findings are that we're able to do kind of some unique things because it is a weekly cross-sectional study. And the first part of that is actually connecting to health systems, a little bit of health systems integration. And so what I mean by that is we have a thing called a protocol 1, and this is when students who complete the survey are actually flagged for those who present with high risk of suicide. And that means an individual who has reported a 12-month suicide plan and has indicated that they're likely to act on that plan within the coming year. And so what we do is for students who are flagged as high risk, they're given an opportunity at the end of the survey to consent and provide their phone number and email to then receive an expedited appointment and referral to counseling. So we kind of help facilitate that and connect the student with counseling services at each respective site. And so at UBC, across the 2.5, a little over 2 years that we've been conducting the survey, 77 students have actually been flagged for high risk of suicide, and around 58% of them, so that's 45 students, have actually been connected to counseling. I will note that we don't actually keep track of what occurs with them after they're connected to counseling. And so that might mean that a student is given kind of an appointment, or they might decline follow-up after that. But we do promise that each student who consents to providing their contact information is connected to counseling within 1 business day. And if they do choose to take an appointment, they're offered an appointment within 5 business days. Something else that the survey is well positioned to do is to actually, the cross-sectional nature and its weekly deployment, actually make it well positioned to analyze trends in unexpected events, such as the COVID-19 pandemic, or even other things such as natural disasters, flooding, things like that. So we can look at kind of how student mental health and substance use is changing with respect to kind of things like that. And so what we see here is we looked at 30-day suicidal ideation across kind of the school year. And so this is starting from March 2019 until May 22nd. And we can see, I want to draw your attention to the kind of two areas where the dotted line dipped below the red line or above the red line. And so that shows significance over the 95% confidence interval curves. And what we can see there is that there's actually no significant association between 30-day suicidal ideation and COVID-19 waves or the pandemic trends, but we do actually find a significant dip in that first half around the summer months, so around May to July, and then an increase in suicidal ideation in February to April, which is around the finals area. So it is more associated with kind of the academic terms and things like that. And so I will close off with saying that, you know, the trends here that we've observed here have actually also been really helpful in informing the development of a non-clinical e-intervention called Minder for Canadian university students. And so you can find out more about that in the posters in the poster gallery. Thank you.
Thank you, Angel. Our last speaker is David Kim, who is a PhD student who's jointly supervised by Rick Procession and Alistair Barr, and is going to be talking about antipsychotics and schizophrenia, doing a meta-analysis. So let's see.
Hello, everyone. My name is David Kim, and I'm a PhD student at the Department of Pharmacology. And today I'm going to be talking about the efficacy of aripiprazole augmentation of non-clozapine dopamine D2 receptor antagonists in schizophrenia, which was studied using a systematic review and a meta-analysis of randomized controlled trials. So antipsychotic polypharmacy is a common practice in the treatment of schizophrenia. However, evidence remains unclear regarding efficacy, especially for the combination of D2 partial agonists like aripiprazole and D2 antagonists other than clozapine. The rationale for focusing on non-clozapine D2 antagonists for this study was that although clozapine is currently the most effective treatment for treatment-resistant schizophrenia, there are a number of barriers to using clozapine. So it is important to study any alternative treatment. So previous studies suggest that augmentation of D2 antagonists, augmentation of non-clozapine D2 antagonists with D2 partial agonists like aripiprazole improve certain side effects, especially hyperprolactinemia, but efficacy remains unclear. So to address this gap, we performed a systematic review and a meta-analysis using major electronic databases, and our inclusion criteria were randomized controlled trials comparing efficacy between non-clozapine D2 antagonist monotherapy and D2 partial agonist augmentation in patients with schizophrenia. So a total of 9 studies, 9 RCTs were included with 791 patients. And 7 of these studies were double-blind RCTs. And aripiprazole was the only D2 partial agonist examined. And as you can see, regarding overall symptom reduction, aripiprazole augmentation was more favorable than D2 antagonist monotherapy. And regarding positive symptoms, which include delusions, hallucinations, and disorganized thinking, there was no significant difference between aripiprazole augmentation and D2 antagonist monotherapy. And lastly, for negative symptoms, which include blunted emotions, lack of motivation, pleasure, social withdrawal, aripiprazole augmentation was more favorable than D2 antagonist, and the effect size is negative 0.5, which indicates a moderate effect size. In conclusion, aripiprazole augmentation of non-clozapine D2 antagonists may be efficacious in patients with schizophrenia. This combination may provide an option when there are barriers to using clozapine, for example, mandatory blood testing, fear of adverse effects, contraindications, or lack of training. And future studies should examine whether the current findings can be generalized to other partial agonists like brexpiprazole and cariprazine, and also whether our findings can be generalized to patients with treatment-resistant schizophrenia. And I'd like to thank my supervisory committee, Dr. Rick Precision, Dr. Alistair Barr, and Dr. William Honer, and also 2022 Psychiatry Research Day. And thank you for listening.
Thank you, David. Okay, so we've got lots of time for questions now and discussion of, I guess, all 5 talks. I also wanted to remind people that on Slido, there's the opportunity to ask questions for those participating virtually. And also, if you just switch tabs, you can go to voting, and then you can vote for the lightning talks and posters. I think you need to do the voting all at once. So also, you know, kind of make a mental note of which lightning talks you liked, and then also go out and see the posters. And then I think after lunch, it would probably be good if you could vote for lightning talks and posters on Slido. And then we'll be able to do the awards later this afternoon. Okay, any questions from the audience? I guess I could start with one for Priya and Carling. It's a really nice data set. So this was all collected locally in Vancouver.
Yes, so this was first at BCMHA, the Burnaby Center for Mental Health and Addiction, so the inpatients there, and then it's now Redfish is the new treatment facility. So it was all inpatients at these treatment centers where we gathered the data.
Was it all interviews, or some of it was sort of surveys that they completed on their own?
Yep, so we always had one research assistant with the participant when they completed it, but it was just one single session of comprising of self-report questionnaires that they completed with the research assistant.
Okay. Just wondering about, I guess, some of the, you know, the childhood neglect and other issues. Is any of that followed up by, I don't know, family history or other, I don't know, other means, like if, I don't know, they were, you know, in, I don't know, juvenile detention or other like indicators of.
Yeah, so there was a lot of different questionnaires that we asked about their history as a child. So we had some questions, for example, on foster care and where they lived as a child, many different factors that might be part of it. And so the main question that we were interested in was childhood trauma, but there was many other data sets that we were asking. And there are some other students in our lab who have some data in the posters here today on some of the other questionnaires that were asked.
Okay, questions of the audience? I don't have my computer. Is there something? Okay, sure, go ahead. Okay, yes.
I have just a sort of follow-up question for Carling. I'm just wondering if you know from the literature or what you would think if the relationship between neglect and sort of this perception about caregiving. Do you think it'll hold true just in mental health populations in general, or do you think it's specific to concurrent, or what are your thoughts about that?
Sorry, just figuring out the microphone. That is an excellent question. And because we were looking at concurrent disorders and we saw the high association between childhood trauma and concurrent disorders, this is what we were interested in looking at. But I do agree that childhood trauma, specifically neglect, might have some effects on association with mental disorders in general, as well as some substance use disorders. And for us, we are not 100% sure like why neglect and not abuse saw these effects, but there are some long-term effects shown of poorer psychosocial functioning for those who have childhood neglect, as well as them perceiving others as less trustworthy, which might be why their experience they perceived to have a poor rating on these subscales for the inpatient consumer survey.
I have a question. And I have a question for Ms. Judy Cheng. I was wondering, you said that you found one of your finds were that young male appears to show deficits in cognitive function during the early age stage. So I was wondering about the older mouse. Did you find the same finding, or was something that they didn't show, didn't appear to have deficit in cognitive flexibility?
Hello, can you hear me? That's a great question. So I didn't have enough time to show all of our data, but you were asking about the cognitive deficits in the older animals?
Specifically the cognitive flexibility that you were evaluating the water team maze. So it was a find that you also find in the older mouses or just in the younger ones?
Okay, sorry. So it's interesting because in the older animals, we didn't see any deficits in cognitive flexibility, but we did see deficits in motor learning, like the training phase of the water teammates for both the males and the females. And I didn't have time to talk about this in my talk, but we actually ran a probe trial between the training and reversal phases where we start the mouse at the opposite end of the maze and see whether they use a more like striatum dependent response learning strategy or a hippocampus dependent place learning strategy to learn the task. So whether they orient themselves in the maze to learn where to turn or using spatial cues in the environment to learn. And so it's only in the response learning animals for our older males that we see a deficit in motor learning. So it's almost like the strategy matters more for the males in learning the teammates task and not as much for the older females. So we didn't see any cognitive flexibility deficits in the older animals, and that was only in the younger males. And we're still working on collecting data for the younger females.
Other questions from oh, Trisha. I'm not, I think, could you try to turn yours off, Judy?
Sorry about that. I was just asking whether for David Kim, if he saw any differences in tolerability or dropout rates between combination therapy and monotherapy in his meta-analysis.
Oh, sorry. No, I haven't. So thank you for your question. And this study only focused on the efficacy and regarding side effect profiles and including dropout rates and discontinuation due to various reasons. I'll be looking at that in my next part of the analysis. Thanks.
Lakshmi? Just a follow-up question to David. Actually, two questions, David. The first question is, I know not all the studies that you included were RCTs. So the question is, did you do a sensitivity analysis just restricting the analysis to RCTs only? So that's the first question. And the second question is, it was surprising to note that when you did the sub-analysis, the efficacy was seen mostly for negative symptoms, but not for positive symptoms. And my recollection of the efficacy data for aripiprazole in terms of its efficacy for negative symptoms is not that great. So just wondered whether you had any thoughts as to why the efficacy was seen for negative symptoms in the meta-analysis.
Thank you for your question. If I understood your first question correctly, my meta-analysis only involved randomized control trials. And so regarding other study types, I did not include in my study. And for your second question, the efficacy was seen only for negative symptoms, but not positive symptoms. And one proposed mechanism is that aripiprazole acts on dopamine D3 receptors, which is emerging hypothesis for the treatment of negative symptoms. So yeah, this one answer for you. Thank you.
Okay, David, do you want to turn your microphone off? Oh, you have one in the back.
Thanks. No, I wanted to follow up on this question and wondered, so there's also the partial agonist effect of aripiprazole, which on its own may not be good for negative symptoms, but when in combination with another antipsychotic may actually prevent the full blockage of the dopamine receptors. I wonder whether that's another possibility.
Yeah, thank you for your question. So yeah, actually when we're only using dopamine blockade, one proposed pathophysiology of negative symptoms is the hypodopaminergic activity of the mesocortical pathway. So when we are adding aripiprazole, which will be acting as a partial agonist, then yeah, that might produce an effect. Yeah.
I have a question for Angel. If I got it right, the survey suggested in COVID that there was a dip in suicide rate. So that's I guess kind of like a term adjusted rate, because I guess different times of year there's more or less stress or around exams or something.
Yeah, I just want to clarify. So it's not suicide rate, but suicide ideation.
Sorry, suicide ideation. I should, yeah.
But yeah, that's correct. So yeah, what we found was that there was an association between kind of more academic terms. So more increases in suicidal ideation when there's more stress surrounding kind of finals, maybe start of the school year, and a decrease across the summer when maybe more students have more time to kind of, relax.
Okay, but the pandemic, if you normalize it to those seasonal things, the pandemic, the start of it was worse or better for suicidal thoughts?
We did see a slight increase at the beginning of the pandemic for suicidal ideation, but it wasn't significant according to our model.
Oh, okay. So the dip down was a seasonal thing.
Yeah.
Okay.
Yeah. I will also note that so our survey started in February of 2020. So we started right out at the beginning, and we added some COVID-19 questions only at the beginning of March. And so we might have missed a little bit of that beginning of the COVID pandemic.
Yeah, I was wondering if, I mean, we definitely heard a lot about challenges, but then there were also, maybe for some people, having I don't know, classes virtually was better and less stressful. And yeah.
Yeah, definitely. We actually have some questions that ask about kind of, living situation and social connectedness about, yeah, during that time period, right, to see if people are feeling kind of more or less connected and if that might affect their stress levels, then that's coming up in future analyses.
Okay, more questions? We've got a few more minutes. I am told the lunch is ready. I guess just one more question for Priya and Carling. I'm just wondering if the data set is going to be publicly available for people to mine, or if there's, I don't know, privacy issues around it, or can it be de-identified and shared? And
From my understanding, I don't believe it is, but that is something I would have to double check with my supervisor to see in the future if it might be made publicly available or not.
Lakshmi.
Yeah, another question for Carling and Priya, I guess. So you both looked at the impact of childhood abuse or neglect on relapse or quality of life, right? So I guess my question is, I mean, there are a number of other factors that might impact those outcomes in that population. And presumably you collected a lot of other data. So the question is, have you looked at impact of multiple factors and found that these were the ones that had the strongest effect, or did you specifically look at this but not other variables?
Yeah, that's a great question. So in my case in particular, because I think both Carling and I did a secondary analysis of existing data. So whatever kinds of questionnaires and assessments were part of like the larger hypothesis, we were kind of limited by what was already collected. And so at least for my purposes, for my thesis, I was just interested in trauma and wanted to kind of narrow down that question to see whether it was related. But you definitely do bring up a good point that there were a lot of other variables that I wasn't able to account for in terms of choosing to see which one would actually have the biggest impact on substance use. But it was more so something that I was interested in exploring just because I've seen other research looking at the effects of trauma and substance use and relapse sort of thing. And I think for Carling might be similar.
And for me as well, there was some demographics that I did look at as well to see if there was a relationship that predicted childhood trauma. And of those, I included those as a covariate in my regression, and there was no difference in the outcomes on childhood neglect on those inpatient consumer survey subscales.
Tricia.
Yeah, I just had another question for Judy. I was just wondering if any of the sex differences you found in your mice, do they seem at all reflective of sex differences in Huntington's seen in humans?
So that's a good question. I believe there isn't a sex difference in terms of prevalence for Huntington's. And Lynn can correct me if I'm wrong, but I believe age of onset tends to be slightly earlier in males, while the acceleration of the pathology seems to be faster in females.
I just had a question for Angel. I was just wondering whether seasonal affective disorders was considered in
Maybe
terms
take
of...
your mask off. Sorry, it's kind of hard to hear.
Yeah, sorry. I was just wondering whether seasonal affective disorders were considered in terms of like suicidal ideation and those trends that we were seeing.
Yeah, thank you for the question. We didn't specifically account for seasonal affective disorder because that's not something that we kind of ask about within the survey itself. But I think it does definitely could provide an explanation for kind of the trends that we're seeing and the dip that kind of occurs, especially across those kind of winter months.
Let's see. Other questions? Oh, yes, Denise.
I have another question for Angel. So in terms of what data you collected, did you collect any qualitative information about maybe potential struggles people were feeling or potentially why suicidal ideation was higher or lower? And was that more particular to school or to the pandemic, or were there trends there?
Yeah, thanks for the question. Our survey itself is mainly kind of quantitative in nature, right? But there are some kind of free response questions. I don't believe that we asked specifically about kind of suicidal ideation and kind of the reasoning why there. But I do think we actually are planning to kind of launch a set of qualitative studies coming out of the survey, and our set of kind of qualitative studies are kind of thinking about targeting specific kind of response types. And so I think that's definitely kind of an avenue that we can pursue, especially in relation to, we also have some questions about kind of treatment seeking and kind of why individuals may not kind of want to seek treatment. And so I think especially amongst this population that's kind of at high risk, especially those who might not think about kind of going to seek treatment, kind of following up with them and kind of talking to them about the reasoning behind that.
Let's see, any other questions? I guess I have one for David Kim. You mentioned this kind of drug cocktail of antipsychotics, and the effect size was 0.5. I'm just wondering, what does that compare to, the effect size? How do you?
Oh, yes, thank you for your question. So 0.5 indicates medium effect size. And so regarding your question about like what that compares to is.
Yeah, is that like standard treatment, or that's with D2 antagonist alone, or how do you? There must be a comparator, I guess.
The comparator would be D2 antagonist monotherapy, and yeah, versus D2 antagonist plus aripiprazole augmentation. Yeah.
But I guess it's not compared back to, and then it might be interesting just for the scale to compare it back to placebo or something, but then that would probably be another set of studies.
Yeah, that would be interesting. For that particular analysis, I would perform a network meta-analysis, and I'll take that into consideration for my purchase.
Because I guess overall, that's probably what's most important.
Yeah.
All right, I don't see any more questions. So I'd like to thank Judy, Priya, Carling, Angel, and David for some really great talks, and just highlighting a lot of really interesting work in the department. And thank you for all the great discussion. So we've had 2 rounds of lightning talks, 10 talks total. And please think about your favorites, and then go and see the posters. Go on to Slido. The link is in the program. And then before around 1:30, I guess, enter your choices, and then press, I don't know, send or whatever. And then you're done. And I guess we're not back until 1:30? 1:30 after lunch. So thanks again, everybody.
Doing the slideshow. I wonder if they want to use this wireless microphone in the afternoon.
I took photos of some of the talks, but yeah, it's just a weird angle, but it's yeah. Yeah, I'm not sure whether to focus on the screen or
This is Martha. She's
In charge of the meeting.
Jessica. She's our 130.
Oh, fantastic, fantastic.
This is Alan, our IT. So
Rolling.
yeah, we thought. And then maybe just if you want to try it now, Alan can probably help you.
Okay.
I think you just have to select the source or whatever.
Okay, perfect. Yeah. All right.
Oh, you're gonna use our own one?
Yeah, if that's okay.
Oh, that is okay.
Okay.
Is she gonna go through Zoom now?
Sorry?
If she uses her laptop, how's it gonna her screen?
is your talk not here on the slide?
It might be better if you can. I don't know about this.
I don't know.
Part.
Yeah.
That's the only thing.
Margaret, is the slide in the show in your slide? No. So you need to connect this one then.
Yeah, so this is a USB. You can actually put the presentation onto the USB that we have here.
Yeah.
Okay,
If
sure.
that's easier.
Yeah, sure.
It might be better. Because
Yeah, because it probably
Okay, so let
take
me,
longer time to
have a
change the
USB?
source.
Yeah, let's take it out. Okay. just a second. Should we close? Should we turn the camera off of the Zoom? I'm not sure if we can do that. I don't know if we can close off because we did that in the morning, like before we started. It was just.
Oh, you mean turn off this, the LC1? I think you do it here.
Are you sure? I don't want to touch this necessarily. I just wanted to, because we want to see it here. We just want to. So which one the host?
Because we are the host, but
The host is right there.
But the share screen is only for this computer.
Yes, LT1. To be fine if you as long as you use that.
Oh my goodness. Okay. We're not going to worry about it.
So your presentation is right here.
Yeah, I have it right here.
Would you be able to transfer to that computer?
Yeah. Do we have a USB? Sorry, I don't. have
No,
one.
but you
one
can
second.
send it
Yeah,
to yourself and.
No, I have it here. Just
Oh,
a second.
okay.
Oh, okay. The other way is using a cloud, like for example, you email yourself, go to your email account and get it back.
Only the easiest way. Yeah. Let's just stick with one.
Like if she plugs it here in the monitor, it doesn't.
No, it doesn't work. The monitor doesn't work. You have to plug from there.
This is a massive.
Yeah.
Yeah. Okay. There we go. And let me just
We don't have a USB key though.
No,
I have
she's
one.
gonna
I'll
eject
Okay,
this one.
but
just wait for
just unplug it. It's fine.
Really?
Yeah, don't worry about it.
Okay. Okay. All right.
Yeah, you cannot wait because you have it opened.
Yeah.
I recognize it.
Take a moment.
So it's someone doing something.
Why is that?
Not us. They're doing something. I don't know.
Oh, okay.
It's not us.
Not affecting us. Okay.
Someone is doing something. from the console that we are there.
Okay.
What's happening? What's
It's
going
full
on?
of surprises.
I
So
know.
the real things happen all the time.
So that
We
is the
didn't even touch
lectern.
it.
Video conference dialing.
I know.
Okay. Sorry, we're just crowding around you.
That's fine.
But because you go to video conference filing,
No, but it's supposed to be on, I think.
I don't know. Still copying.
Okay.
It's a big file.
Yeah, I have a couple videos, so hopefully.
Oh, couple video. Okay, so we better use a slide then. This is Microsoft, right? Your presentation.
Yeah, it's Microsoft PowerPoint.
Okay, good. When you present, the mic will be here, so.
Okay.
And you use this device. This is the laser pointer. This is advancing, going back.
Is everyone okay? Alan, you're gonna be here for now?
Yeah.
Yeah,
Go ahead.
So it's just a matter of putting up the
I just plug a USB key back in there. man.
yeah, and
But-
pull up the lunch slides. Yeah. I
Mhm.
guess. Okay, perfect.
Go ahead, go ahead. So where do you put the file then? Where you need to put her file in there?
No, she's putting it in the USB and this USB goes back here.
Okay, I know. Let's take a look. There we are.
There you go.
Cut this.
Yep.
It's okay? Okay.
Okay, try to open this for me.
So.
Excuse me. Not there yet.
There we are. Okay.
It's automatic.
Okay.
So which one is that?
So this is you, Jessica Russell?
Yep.
Okay.
No, not this one. This one is.
No, she wants to, you wanted to test, right?
No, this one.
Oh, sorry. No, I mean,
You open the other one. That is this one, Jessica, right?
oh. Oh,
Yeah, you
sorry.
open the one that is not supposed to be open.
Oh, I see. Okay.
Yeah. Oh,
Escape.
let's close this first. Okay, now we open it.
Okay.
That's better. Because it's got video. I don't know why that file is there. Because maybe it's the Apple and they copy something else there too. Oh, this take a long time to open.
Yeah, it's okay. It's
Almost there. Wow.
there you are.
Big
Okay.
file.
Do
Go
you
ahead,
wanna
try it.
test?
So
Maybe the play your video. That is,
Yeah, so you hit the slideshow, okay, and then you use the clicker to move your slides along. See, it shows there,
Let's see the
and then
video.
you're projected here.
okay.
Yeah.
Yeah, so let's see the video is working.
Oh, okay.
Oh, here, hold on, let's go back.
Is it working?
No.
Oh.
Is there a way that, can I click it?
Yeah,
like this?
you need to play it. Click the play button.
Oh.
Is it working?
No.
Oh.
Can I play?
Quick time not available, it says.
Huh?
Quick time not available.
Quick time is not available. Oh, unfortunately, you need to call this computer doesn't belong to us.
Oh, I see. Okay.
I wonder if my other video will play then. No, it's not playing.
Oh,
on
this
here.
one works.
No, it's not. That's not what it should look like.
Oh.
It's
Oh.
all screwed up.
Yeah, video is tough sometimes. Like,
How did Margaret play the other one? Did she have? So it's only QuickTime that plays this, or like can there not be another software or?
too late for now. Like, if it doesn't work here.
That's another of the videos were.
Okay.
Because like, we don't have rights to this computer to do anything anyway. I tried to do something else, I couldn't even do it.
I see.
Yeah, they have policy
Is
on
this
this.
another video supposedly or?
Yeah, there's two and neither work.
Oh, okay.
Neither video works.
Oh dear. I wonder why.
But it works on your computer, right?
Yeah, and I know if I connect, it'll work, but then they have the problem with Zoom.
Oh, I see.
But the problem is that if you connected your computer, you're not sure you will show it to the Zoom patient.
Yeah, yeah, that's what
Zoom
Yeah, that's what she's saying.
audience, right? So.
Uh, is it risky if we try to connect to her laptop?
Yeah, we can try to connect it to a laptop if you want.
But it might be. Meantime, I will turn this off.
Video conferencing. How come it's video conferencing?
Yeah, it's supposed. I think it's supposed to be complete.
Return, but how come this thing is up?
I have no idea.
I don't want to mess around with this.
I know.
Lantern PC. So which one is the PC? This is the podium camera, right?
Yeah, that's the podium camera. That's the one right above
Okay,
us.
then
And
we
this
need
one
to
is the
choose this
lantern
source. Lantern PC.
HDMI?
Yeah. Try HDMI. Okay, then we gonna go for let's try HDMI.
On HDMI, okay.
Yeah.
Great. Okay.
So, but I'm not sure the Zoom people see. 1 second.
Yeah, the video works.
Can you see, Alan?
Yeah, the video is playing.
But yeah, can you see it on the Zoom? Okay. Okay, great.
And that video is playing too.
So we need to tap. Are you the first one?
Yes.
Okay, we just leave it like this.
Okay.
Yeah, you can just leave it here.
Okay.
My computer.
Yeah. Or just.
Or do you want?
Don't worry. You don't need to leave it here. You just plug it in when you're here. Okay.
Yeah. Okay. Yeah.
Yeah, but yeah, can you be?
I'm gonna go change it back to the Lantern PC.
Okay.
I was gonna say I've done this before where they've been able to connect me via Zoom, but I don't know how, so I'm glad you were able to figure it out.
Don't worry, it's okay. I'm great you came.
I
know, I see you'll be setting up while people looking at you, all of us.
Okay, perfect. Thanks. I'll
Hey, see you later.
a little bit.
Yeah. Go get some food.
Where is the lunch break?
What are you doing?
Lunch break. I'm
You're doing the poster?
Yeah, just the lunch breaks there. Okay. Okay. Okay, let's go eat.
Nah, I don't eat. Let's go. I want to go outside someday.
not here. He has to bring his own
Yeah.
either his device or his lines.
Well, we just help him on the spot then.
Huh?
Just help him on the spot. It's easy anyway. You plug in his USB key, like Dr. say, and play from there.
Okay.
Yeah. So we feel that the connection works. Then you have to choose like the HDMI on the left hand. Then it will show on the screen. I didn't go to the slido.com at all.
What, sorry?
The Slido. I didn't touch the Slido. I think that they Dr. Murphy and Dr. from who is taking care of. I'm not paying attention. Are you married? You have a husband, right?
What?
You have a husband, right?
I do.
So are you getting any kids?
I want to, yeah.
Or you planning to?
Planning to, yeah.
Okay. Yeah. Because I wasn't planning to have kids at all at the beginning. And the next thing is like, oh, okay, fine. And then after that, I think I'm really happy that I have a kid.
Oh, really?
It's totally different. Yes, after you
You have one. How many do you have? One? One, okay.
Yeah, we almost lost him. Like, because we doing it in Women's Hospital. And luckily, my wife didn't send home. And that day is like our doctor, like the old I forgot the name, whatever doctor take care of women. But he was there. He was there it's his turn to do the walk. So the student asked my wife to go home, but our OB insists she has to stay. Otherwise, we lost the baby. Because we have my wife has a placenta eruption. That means the support system fell off. So blood is everywhere. And then
good morning.
And then I have to help to remove the whatever instrument wire.
Oh my God.
Yeah, and the base.
cord?
Yeah.
You mean the cord?
Yeah, whatever it is, like I have to help to remove. They have to go into operation room right away.
Oh my God.
Yeah, there's our doctor left already. It's funny when he left. Oh, we'll see you tomorrow, and then your baby will be out tomorrow. And then next thing is like my wife fainted because lost too many blood. And then I was accompanied by a nurse. Like this needs to me, make sure I'm not go crazy. And then my wife is in the operation room last minute, and they cut wide open, take the baby out, and the baby is they have a score 0 to 9. My baby is only 1.
Oh my goodness.
0 is dead.
Oh.
So they do a resuscitation for almost 5 to 10 minutes and got my baby back. Got my boy back. We are in ICU, like a newborn ICU for 5 days.
Oh,
Yeah.
but now, oh wow.
And my wife was, it's funny when the baby is out, the nurse is asking me like, you want to stay with the baby or stay with my wife? And I asked her, is the baby okay? Yeah, it's okay. Never mind, I go see my wife. And then my wife was shaking, no matter how many blankets put on her. She's still shaking because she lost so many blood.
I know. Oh my God.
Yeah, and then like, I think within 2 days, it's keep taking painkillers. And then within 2 days, we kind of get her in a wheelchair and go see the baby. She doesn't want to like. I think we have to push the bed down there. Yeah, because you want to see the baby.
Yeah.
Yeah, it's not an easy one.
Was there any complications? That's why, or
Percentile eruption. It's only 1 in 200 chances you will have that.
Oh, wow.
Yeah.
Intense.
And the baby may have a chance of dying.
Yeah, maybe I'll move over
And-
there. Because I don't think I can use Margaret's computer anyway. As password.
What password? Margaret password?
Yeah. Because yeah, she said to take photos of the speaker and of the.
I don't know.
The Slido, the faculty members doing it, right? The Slido. Like,
Yeah. We don't need to care about slide.
okay. Say so.
Yeah, you take care of the whole.
Yeah, I mean, I'm not supposed to, yeah.
Don't lose your hope. No, I think they killed this section. I'm not sure what this is. Anyway, this is the most important one.
Yeah, no, what happened is when he used the host, he made you the host, I hit mute all right away. And you're not supposed to do, I did that.
And what we supposed to do? Which one we should do that?
What we should do is to mute everyone individually.
Yeah, that's what should be told me.
Well, yeah.
Over the phone. Over there when I have no phones. I didn't pay attention. It says like you just go into that person and mute it. That's what he said. It doesn't say it cannot be wrong.
Oh, when was that? What was that?
No, when we are having problem. When we are having problem, when we start noisy. And during the break, before the next session, the middle break. And then someone's talking after because the meeting is done, right?
Yeah.
So we start hearing noises, and then we have a unknown host at that time. So and I call them, and he just say, oh, go into that guy, look for the host, and go into that guy, and mute that guy. And then I couldn't find the host.
I know, we can find it. It turns out that's the host.
Really? Oh yeah, because they take over.
Yeah.
So the control is in the control room. Nothing we can do. So it's a mix up basically.
Because we had the drive right here a week ago. Yeah, we just went to see.
Oh, you got a drive on. I didn't know that. That's what. I have no idea you guys are like totally in charge and otherwise.
Oh no, look at us. We were all in actual charge. we all fell apart. Because something,
Yeah, because I have no idea what I should do when I keep it. Well, anyway, it should be to help.
No, you're very helpful.
Because I had experience with a lot with the Zoom though. I have experience with like on site, everything. I know how to do
Zoom. I know how to do this, but to do them all together is much.
Oh, it's so easy.
Yeah.
Well, the best way is if you run into problem, call them right away. Because you cannot guess, basically. The first thing you do, call them. I thought it's like, moderate.
Just moderate.
Only 2 talks, that's it. Yeah, not much. Yeah, it's easier compared to the morning. But I'm glad you guys, one thing I like the arrangement is to put all the slides inside.
Oh, that's Margaret's work.
This is really good.
Yeah.
The basically is in one big PowerPoint.
Yeah.
Oh, cool.
Cool. Hey.
Yeah, we went into the problem of the host, like one time when we during the middle break, someone is talking behind.
No, we didn't realize that this computer is the host, and we were trying to figure out.
How to get into the host.
I made actually the computer is not the host, it's the codec. So
Is the console?
Yeah.
Oh, that's
Yeah,
right.
yeah,
Because someone is talking behind, and I call them. I couldn't mute the guy. He told me you just go to the host and mute it. I said, and then he start looking for the host. There's no host.
Yeah. Okay, so somebody joined this talk.
Somebody joined, and then the microphone is on.
Okay.
And then there's all of a sudden there's a late woman's voice, there's a man's voice.
Yeah, I think we just don't have that ability to do all. So
Yeah, our rooms don't have that capability because in case this room disconnects and joins back in, like for half a second, it'll join unit and then it'll be a problem. So we don't have that capability. So
I think it's another point called
yeah.
we created, and then next thing is all fine.
We're host at the host now, but they have to connect the room.
Yeah. So
It's okay now. Okay, perfect. And then in the afternoon, there's a connection tested already. Okay. So I think. Okay, perfect. Yeah. Thanks for coming back out. Of course, I'll come back at 1:30 just to make sure that's when you guys will start. Yeah, we got need to connect our laptop to the. Oh, you are? Yeah. Okay. Where you show you? We try already. Oh, okay. Yeah, because they have video that cannot play on the other PowerPoint that we. So yeah, anyway, it's good. Thank you so much. Absolutely. Yeah. 20 years. This is topic called almost 40 years.
Oh, okay.
This double mine.
Yeah, it's double yours indeed.
This is the longest. I think it's one of the longest. One of the longest. Not too many. Oh, yours is what? He put the last one in from the paper. He have multiple of them. All in frames, except the last one he don't bother to go. And they send it to him, he just squeezes somewhere. It's so funny. Yeah.
Like the first one, if it's 10 years or something? Or?
Yeah. I think the first one is 25 years. I never get one. I never go to a dinner. If you are 25 years, you are entitled to go to a dinner. And you need a partner to go with.
Wow, so you're not even qualified yet?
No.
Wow.
Well, 5 more years. I retire already.
Well, stay on just for the dinner.
Actually, I can retire now. I can do early retirement. I think Colin Pina is retired.
Colin?
Yeah, Colin Pina.
Ah, Colin Pina.
Yeah, our previous guy. Hello.
Hey, do you have an extra slide?
Yeah. Dr. Vila.
Yeah.
I usually teach here
Oh, so
for
you know how.
med school,
Help
so
yourself.
I believe I know my way around a little bit. So I can troubleshoot a little bit. It's the HDMI. I only have to remember my password.
I think you just choose this.
HDMI, yeah.
That's it.
It is. It is
Oh,
talking.
I saw your picture now. The lightning. You like to use Mac or PC more?
Mac.
You like Mac?
Yeah.
Why?
You won't believe this, but I grew up with Mac, actually.
Oh.
And the reason is my dad is a journalist, and it turns out that Mac computers from the very beginning use the system with Windows and stuff.
Yes.
Way before
the
Microsoft. Exactly.
The guy who's process.
That's fine. Hi, hello, everybody. And so everybody can see my
Actually, we can
slides.
turn off the mic.
So yeah, it seems that it's turning on.
There you go.
So
Nobody can hear you now.
So yeah, I did grow up with Macs around the house from early on, and then eventually I did have a PC at some point, but
Docs?
I switched.
DOS? You have a DOS? DOS, DOS.
Yes.
DOS PC. Yeah.
Yes, and we had also an IBM 512.
Yeah, I use the 5.25-inch copy.
Yeah. So they work.
Good. Yeah, it works.
See you later. I'm gonna see.
Yeah. All right. You're one of the judge?
What's that?
Are you the judge?
No.
Yeah, the presenter behind you test his MacBook already too.
Oh, okay.
Yeah, he's also using HTML.
Okay, great.
Yeah, so you both will be a Mac presentation. You don't even need to let the PC, the Lantern PC anymore. All right, let's use this.
Perfect. Thank you so much.
You're welcome. Do you want to wear, like, do you want to just use this microphone or
Yeah, I
you
can
want
probably
to?
just use that microphone.
Okay. It's mute right now. I think Dr. Frangu will take care of it. Video, audio source. So that's how to turn on.
Testing, 1, 2, 3, 4. Basically, you can stand further. If you will adjust the volume a bit, you can stand here and then they can hear you anyway.
Okay, perfect.
Right now it's mute.
Got it.
I'm ready to go.
Let's get it started.
Exactly. I think because we have it connected here, they have it showing my slide right over here. It's the same, it's the same file. How do you think there's people coming in? Because to be aware. Right. I tried to make it very simplified as much as I could. Yeah. Exactly. Maybe during the. Coming up. For sure. Yeah. I was just going through the points for the basic.
I just interrupt. I wanted to say that I came and I tested mine already.
About the question.
Okay.
And I think I will introduce my children use this again. And then look at question, the one here, but how they can take them yourself.
Okay.
Yeah, you point to whoever it is. And I would just read, is there any questions like?
Like, so we take questions
At the end.
Yeah. Perfect. Yeah.
So when you finish, you take questions, right? If there are any questions, slide in the very quiet. If there are any. So I will. Just be mindful of the time.
I try to control my speaker balloon time, like putting slides.
Yes. ask you something? Dr. Rosen is presenting from her laptop. Do we need to connect you to this? So do you want to come to this part? So Dr. Rosen has her presentation on her laptop. But it's up there, and I believe it's presenting on it's just because Zoom. Yeah, it's just because it's taking it from your laptop. Yeah, but it is linked already. Yes, yeah, they checked that. Okay, you checked. Oh, okay. Yeah. Okay. No problem. And then I think you need to talk. You gotta unmute. No, okay. Are there many people still outside, or have most people come back in? Do you know? I think sort of Margaret and Co are doing that. Okay. But I was wondering, shall I start or shall I wait a little bit? If we should. 5 minutes later now.
I kind of like that. So welcome to the afternoon session of our research day. And we will have two talks, one preclinical and one clinical, to sort of highlight the sort of the dual pathways of research within the department. There will be questions at the end of each presentation that will be handled by the presenters themselves, and I will help them with any questions that they are in Slido. So our first presenter for the preclinical section is Dr. Jessica Rosin. She's a new recruit to UBC, having come to us in 2021 from the University of Calgary. Her lab is focused on examining immune regulation of developmental programs, and specifically on how gestational perturbations, such as maternal stress, can impact fetal development. This line of work builds on his pioneering findings about the heterogeneity of the population of microglia in the embryonic hypothalamus, and the fact that their constitution is altered in a sexually dimorphic way following gestational stress. As the title shows, today she will focus specifically on fetal microglia and sex-specific neurodevelopmental responses to maternal stress. And as we heard from the previous thing, sort of early adversity is one of the key predictors of long-term outcomes. And it's very nice to be able to use animal models to experiment and understand a little bit better how this happens and extrapolate for that in humans. With that, I would like to welcome Dr. Rosin.
Well, thank you for the introduction, and I want to thank the organizers for giving me the opportunity to speak to you today. So I'd like to start with this image, and basically this was on the cover of Times around 10 years or so ago, and it's really at the time that we began to realize that in addition to genes and environmental interactions, the actual environment that the fetus is exposed to during pregnancy can have a huge impact on a number of different developmental programs, both within the central nervous system and outside of it. So with regard to different perturbations, we could be looking at cancer, diabetes, Alzheimer's, and a number of other different disruptions to the developing fetus. And so how we kind of look at this is there are some things that we are really well aware of that during the gestational period that you can be exposed to that can have a detrimental effect, such as alcohol, certain drugs, smoking, and trauma, infection, but there are also certain other chemicals and endocrine disrupting chemicals such as bisphenol A, for example, that you might have heard of that can be detrimental to the development of the fetus. And then there are other things that aren't necessarily detrimental to the fetus, but we need the appropriate amount of balance. So diet, exercise, sleep, these are all things that if you get a little bit too much or a little bit too little of can perturb the development of the fetus. And today what I'm going to be talking about is stress. And so I just wanted to point out that while we did a lot of these studies prior to the start of the pandemic, there have been a number of studies during the pandemic that have actually shown that COVID-19 pandemic has taken a significant toll on the mental health of moms. And it actually was shown in a report that was done at the University of Calgary while I was there, that anxiety and depression in moms, for example, nearly doubled as a result of the pandemic. And so there's this heightened level of stress, anxiety, depression in both pregnant women and also moms that have young children. And so the region of the brain that we study is the hypothalamus. And so it's something that's not often talked about. And that's because it's at the base of the brain. And basically it's normally just thought of, okay, this is a brain region that's involved in homeostasis. And so heart rate, blood pressure, those type of different controls, but also it's involved in addition to those type of regulatory systems. So temperature control, it also is involved in hunger and satiety, thirst, your circadian rhythm, so your sleep, your parenting behaviors, reproduction, social behaviors, mood. So it has a wide variety of different controls. And it's this real functional link between our central nervous system and our endocrine system. And so what's really nice is the hypothalamus is comprised of almost a dozen different nuclei, depending on how you determine the rostrocaudal axis and divide them. But what's really nice is we use mice as a model, and these different nuclei within the human hypothalamus are really well conserved in both their localization, the neurons that are present, and their functions. And so using mice as a model, we wanted to really study how stress from mom is transmitted to the fetus and how it might be disrupting the developmental programs within the developing hypothalamus. And so right now, my talk is not going to be about what the signals are that mom is receiving that are stressful, that are being transmitted to the fetus, because we don't know what that is at the moment. It could be through cortisol, it could be through a number of different other factors that are transmitted through the placenta, but it's more this idea of what's going on within the developing hypothalamic region of the brain in the fetus. And so we have these neural stem cells that go on to make the neurons, the oligodendrocytes, and the astrocytes within the hypothalamus, similar to other brain regions. And so it could be that this stress signal that's coming from mom is being sensed by the neural stem cells and directly disrupting the development of neurons or their specific subtypes, the numbers, the localization, something like that. Or it could be directly being sensed by these neurons that are being born during embryogenesis and pregnancy. But what's also could be happening is during this time point, during fetal development, there are also these immune cells that I'll talk to you about on the next slide in a minute here and introduce them, that are called microglia. And so these microglia are within the brain, and they're these little sensors of what's going on in the environment. And so what could be happening is the microglia themselves could actually be sensing this stress signal. And what's important is that we're actually learning that microglia can communicate to the neural stem cells. They can also receive signals from the neural stem cells, and they can also signal to or receive signals from neurons themselves. And so they might be this underappreciated sensor of these different environmental perturbations during the period of pregnancy. And so, as I mentioned, microglia are these resident macrophages, these immune cells that are phagocytic within the developing central nervous system. They originate from erythromyeloid progenitors within the yolk sac, which is actually very unique. And so they go through this intermediate macrophage stage, and then they go right into the developing central nervous system. And it's both the unique origin from these erythromyeloid progenitors from the yolk sac, and also the environment that they're exposed to, so the central nervous system, that allows them to become these unique cells, these microglia, because other cells in other macrophage type cells within our body usually come from hematopoiesis and a monocyte precursor cell. And so they have this unique origin, this unique localization. And while it was originally thought that they were these kind of cells that were just waiting around for the development of central nervous system, and they were going to play a role in immune surveillance and synaptic pruning and all these postnatal and adult events, what we're actually showing, we and others, is that they play a very prominent role during development of the brain. And so here's just a video that is actually showing their activity. So these are embryonic microglia that we've done live cell imaging on, and you can see that they move around and they reach out and they respond to different signals, just as you would see in the adult brain. And so the only difference is they're a little bit more amoeboid. The processes aren't as ramified, so they don't look like these star-like cells that you often see in the adult, but they are contributing to the development of the central nervous system. And so our question really became to determine if these fetal hypothalamic microglia respond to maternal stress and whether this has an impact on neural development. And so we know that the hypothalamus is involved in homeostasis. We know that microglia are involved in surveilling and basically making sure that there's normal homeostasis within the brain. We know the hypothalamus is involved with the stress response and fight or flight. And so the idea is potentially these microglia are these sensors of some sort of stress, and then they're communicating it to these neighboring cells within the developing hypothalamus and potentially perturbing their programs. And so really to start, we had to go through the hypothalamus. So this is an image through a cleared embryonic day 15.5 brain along the rostrocaudal axis, looking coronally. And basically we're looking at all the microglia within the hypothalamus, and this is just it overlaid and pseudo-coloring them in different planes along the rostrocaudal axis. And what we began to realize is that there appear to be these unique subpopulations of microglia in the developing embryonic hypothalamus. And so what I'm saying here is that we have populations that are lining the third ventricle, and that's where all of the neural stem cells are located within the developing hypothalamus. So we have the opening, which is the third ventricle right here. And so we have a bunch of them that are very amoeboid, so they're very circular, and they're residing right beside that neural stem cell niche. And then we have a number of other populations that are localizing near different nuclear structures. And so it seemed that we really had to understand what the different types of microglia during embryogenesis were in order to understand how they might be perturbed by maternal stress. And so what we decided to do was to collaborate with a colleague at the University of Calgary, Jeff Bernasky, and a really talented PhD student at the time, Sartek Sinha. And what we did is we took our CD1 mice, so just regular wild type CD1 mice, and we either collected embryonic day 15.5 male embryos or female embryos. And then we also collected those embryos that were exposed to maternal stress. And the stress paradigm we decided to use was cold stress. So the hypothalamus responds to temperature, but we also used this stress because we found it was a more mild stress in comparison to some of the other sleep deprivation or restraint stress models that are out there that can often result in embryonic lethality, or some of these more severe models that we didn't want to get this very severe model. We wanted to take more of a mild stressor type of phenomena. So they were exposed to 4 degrees Celsius